So 3, 2, 1. We are live now.
Good evening, everyone, and welcome to the iHera series. And today's theme is Oocyte development, the beginning of play after pause. And to have the conveners of this webinar, we have with us Dr. Ved Prakash, Dr. Sanjay Shukla, and Dr. Charu Dutt Joshi. I will now read out the CVs of the coordinators. We have with us Dr. Nidhi Singh. She is the medical director of Tejasvi Women's Wellness and Maternity Center, Varanasi. She is a senior consultant embryologist at Panacea Hospital, Varanasi. We have with us Dr. Yoshita Tanwar. She is currently working as a clinical embryologist at Oasis Fertility Center, Bangalore. She has worked as a senior embryologist and lab in charge at New Life India Fertility Center, Delhi. We have with us Dr. Nancy Sharma. She is a senior consultant embryologist, Department of Reproductive Medicine, Jindal IVF and Sant Memorial Nursing Home. And we have with us Dr. B. Ananta Lakshmi, who is a consultant and senior clinical embryologist in the Janani Trichy Fertility Center. Now I would like to hand over the session to Dr. Sharma. Ma'am, over to you, please.
Now I would like to call our conveners on the board, please. We have with us Dr. Ved Prakash. Sir is consultant clinical embryologist, scientific director, Sperm Life Advanced Embryology Lab. He was also a past president of ACE India. Then we have with us Dr. Professor Sanjay Shukla. Sir is senior consultant clinical embryologist.
Next slide, please.
Have the CV, please. Sir is Senior Consultant Clinical Embryologist, Adjunct Professor Clinical Embryology, Department of Reproductive Medicine and Surgery, Mahatma Gandhi University of Medical Sciences. He is working as the past president of ACE and chairman of academic wing ACE. Now we have with us Dr. Charu Dutt Joshi. Sir is medical director of Gene India ART Bank. He is also lab director of IVF unit of RD Gardi Medical College. And is co-founder of iHera. Now I would like to call Dr. Nisha. I would like to call Dr. Sanket to introduce Dr. Nisha and Dr. Vandana. Over to you, Sanket, sir.
Yeah, sorry for the very good evening to all. Firstly, warm welcome from iHera. We have iHera, and I think Dr. Nisha has not joined us for whatever the reason. So,
Sanket, Dr. Nisha is there.
Okay, well, cool.
I think,
So Dr. Nisha Bhatnagar is MBBS and MD from Afghan. She's currently working as medical director at IVF Fertility and IVF Center, New Delhi. And coming to her education and training, she's a senior research associate on the ICMR project titled on effect of yoga and counseling on the outcomes of pregnancy in patients undergoing IUI and IVF, and at IVF Center at Maulana Azad Medical College, Lok Naik Hospital, New Delhi. And also she has completed her MD at Dr. Baba Saheb Ambedkar University, Aurangabad, and her MBBS from the same university. And areas of specialization of her will be natural cycle IVF, and as well as minimal stimulation IVF, as well as poor ovarian reserve, as well as infertility management. She has memberships at various reproductive medicine societies, like starting from IFS and FOXY, ISAR, and ASRM. And she has presented a paper at medical management of ectopic pregnancy with respect to their experience at AICVG Agra in January 2004. And she has also publications on various publications on different journals. So welcome, Dr. Nisha. And I would like to request you to introduce the first speaker of today, Dr. Vandana Hegde. And over to you. Nisha ma'am, are you there? Well,
tell Dr. Charu he can do the introduction.
Charu sir, please introduce madam.
Very good evening to all. It's my privilege to introduce Dr. Vandana Hegde. She is presently Clinical Director, Hegde Fertility Unit of Hegde Hospital. She has been awarded for standing first in final MBBS exam, adjudged outgoing student of the year. She has done MS Obs Gynae, postdoctoral fellowship in reproductive medicine, diploma in reproductive medicine and embryology, member of ASRM, ESAR, FOXY and IFS societies. She has been the faculty for ESAR, ASICOG in class, awarded first prize for presentation at UA FOXY 2005. The center has received multiple awards for excellence in fertility care. So been in the field of reproductive medicine since 2007, established Hegde Fertility Center in 2010 at Hyderabad with 4 branches in the city, and presently running a 1-year full-time program for fellowship in ART since last 3 years. So over to you, madam. Welcome, and waiting to listen a good outcome of the topic. Thank you.
I would like to thank Ahira for this wonderful opportunity to be among all the embryologists here today, and also to the conveners, Dr. Ved Prakash, Sanjay Shukla, and Charu Dutt Joshi. Thank you for the warm welcome. So the topic given to me today is the beginning of the largest human cell, that is the oogenesis. I'll just share my screen. It's seen, right?
Yeah, ma'am. Please go to slideshow mode, ma'am.
Yeah. One second. Yeah. So oogenesis, yeah. So what I would say is I'm a clinician at the end of the day. So I will be bringing across the topic in a clinical point of view, because I know that the further topics are all related to the field of embryology. So as we all know, the oogenesis, if you talk, we should always start with the ovarian reserve. And this is something that we are all aware today, that ovarian reserve is what we are all worried about today. And that's something that is just goes on depleting from the fetal life to the age of menopause. So what starts with 6 to 7 million oocytes during the fetal life of 3 to 7 months, which drastically by birth it is reduced to 1 to 2 million, by menarche to 300,000, and it's only hardly 500 oocytes that ovulate in a woman's lifetime, and by menopause it is around 1,000 follicles. So if you look at oogenesis, what is it? It's a type of gametogenesis through which the ova, which is the female gamete, are formed, and the produced female gamete is known as an ovum. And it occurs in the outermost layer of the ovary, that is the cortical layer. And these are the step of oogenesis, which starts as the oogonium, which is a stem cell, and which undergoes mitosis to produce the large number of daughter oogonia, and which passes through various phases. There is the primary oocyte, the secondary oocyte, and finally it goes through the ovulation process and fertilization. So if you look at the oogenesis, which starts at the fetal life, the oogenesis is actually a protracted process that encompasses the birth, growth, and maturation of this cell, which is unique in its ability to propagate another generation of organisms. And the maturation of these oocytes begins even before birth. If you look at the primordial germ cells, they come to the gonad of the genetic female and differentiate into oogonia, and they undergo a number of mitotic divisions. And by the end of the 3rd month, they are surrounded by a layer of flat epithelial cells. And this layer of flat epithelial cell is actually derived from the surface epithelium of the ovary. Together, these two constitute what we call as the follicle. And we need to always realize that as a clinician, we only see the follicle. So what we want to correlate is how the follicle development correlates with the oocyte maturity, because that is how we can tap the potential for the best pregnancy rates in our own patients. So if you look at the ovarian follicles, these are small fluid-filled structures in the ovary that house the developing oocyte, and they go through the primordial follicle to the primary and secondary follicle and the Graafian follicle. So the figure here shows the development of the antrum. Now, why antrum is very important? Because it is only when this antrum develops, which is a fluid-filled follicle, it becomes visible on the ultrasound. So the antral follicle stage is where we see on the cycle day 2, and then we are able to count the number of antral follicles to estimate the ovarian reserve, and that is how why we take a lot of clinical decisions on ovarian stimulation. Now coming to the oogenesis, what happens now in the fetal life? Now the majority of the oogonia, they continue to divide by mitosis, but some of them, they arrest their cell division in the prophase 1 of meiosis 1 and they form the primary oocytes. Now during the next few months, these oogonia, they rapidly increase in number, and by the 5th month of prenatal development, they reach the maximum of 7 million. And now consequently, the death also starts. As the cell death starts, the many oogonia and even the primary oocytes also can become atretic, and they usually stable around a number of 500 to 700,000 oocytes by birth. Now at birth, all the surviving primary oocytes have entered prophase of meiosis 1, and most of them are individually surrounded by the layer of flat epithelial cells, where what we now call the primary oocyte. And now this maturation of oocytes are all there until the age of puberty. Now what happens here is that the primary oocytes, they have started the prophase 1 of meiosis 1, but instead of proceeding into the metaphase, they enter a diplotin stage, which is the resting stage during prophase, and this is characterized by a lacy network of chromatin. So this is aptly the name that you've given for your CME today. It's the pause over here. So now the primary oocytes, they remain in prophase and they do not finish their first meiotic division before the puberty is reached. And this is a very long, there's a lot of chemicals involved over here, but we give the name of oocyte maturation inhibitor, which is a substance which is secreted by the follicular cells itself, which keeps them arrested in this phase. And during childhood, most of these oocytes become atretic, and it is only approximately 400,000 which are present at the time of puberty, and it is less than 500 which are going to be ovulated. Some oocytes that reach maturity late in life, if you look at patients now trying to conceive at 35 to 40 years, you can actually think that all these follicles have been in the dormant stage, that is the diplotin stage, for nearly 40 years or more before ovulation. So this prolonged exposure for such a long duration could be one of the reasons that these oocytes are vulnerable to all the damage, and thus resulting in the risk for chromosomal abnormalities in the children. Now, what controls this folliculogenesis? Now, in the clinical point of view, the control is mainly the hypothalamo-pituitary and the ovarian axis. And this entire process is the endocrine control, which actually starts, the endocrine control starts only at a later part, that is after the secondary oocyte. Now, this mainly functions to the positive and negative feedback mechanism, where the major hormones involved are the GnRH, which comes from the hypothalamus, the pituitary hormones, the FSH and the LH, and then you have ovarian steroidogenesis in the form of estrogen and progesterone. Now, it is just not these endocrine, but even thyroid, prolactin, they all have a significant role. So this is the final cascade that we see, which is very rhythmic, I would say. So it is the hypothalamus which gives the signal to the pituitary to release the FSH, which starts the follicular growth, and then LH comes mainly at the time of ovulation, followed by, so you can see the progress of the ovarian follicle over here, starting from the antral follicle, growing into the Graafian follicle. At the time of ovulation, the ovum is released and followed by the corpus luteum. So the role of FSH mainly is for the development of the growing follicle, but at the same time, it is also equally important for the steroidogenesis. We talk a lot about LH because when it comes to oogenesis, we are all worried about the maturity of the oocyte. And we know that LH is finally the signal that is important for ovulation, which times the ovulation, and thus we associate it mainly as the most important hormone for oocyte maturity. But we should also understand that when it comes to LH today, we are recognizing more and more its role in early folliculogenesis. So the end product is as good as the foundation that you start with, that's the basal. So here what we have realized today is the LH is very important in the initial part of folliculogenesis because it provides the substrate, the main androgens, which actually brings about the FSH receptor expression in the follicular wall, and that causes the signaling pathway for the attachment of the FSH into the follicular for folliculogenesis also. So its role in folliculogenesis and ovulation is both accepted today, and its role in steroidogenesis. As I told, LH is very important for the oocyte competence and maturity. Now, why? Because it's very important to have the optical follicular development to ensure the oocyte competence. Here, the LH window concept is what we have to understand, that very high levels and very low levels, we can land up with problems with the oocyte quality. Now, very low levels, what will happen? You will have impaired follicular growth, impaired oocyte maturation, and inadequate steroidogenesis of androgen and estrogen. And even if you have an excess high level of LH, LH could lead to follicular atresia, oocyte development gets compromised, there is suppression of the granulosa cell proliferation, and sometimes even premature luteinization. So what we call is the LH window, which should usually be between a threshold of 1.1 and below a ceiling level of 5. So this is the ideal LH that we maintain during the folliculogenesis. Now, if you look at the hormonal control of folliculogenesis, I told you this is a gonadotropin dependent phase and the gonadotropin independent phase. So it is from the probably day 21, that is a week before the menstrual cycle, that is where the gonadotropin dependence starts. So usually you have a phase of recruitment. So if you look at it, the follicle starts from the primordial follicle, and then there's the primary and secondary oocyte. Now these stages are all gonadotropin independent. And if you look at the AMH, it actually indicates which is one of the best, highly sensitive among all the ovarian reserve tests available. It still only depicts the ovarian reserve that starts from the primary oocyte level. The AMH does not reflect the primordial follicular pool. So the gonadotropin independent is mainly important for the recruitment of the follicle, which is actually a phase lasts at about 60 to 90 days. And finally, the selection process starts probably a week before the onset of menses, where there is a phase of selection of the oocyte. And then there is the growth of dominance, that is the selection and dominance happens. Once the dominant follicle is selected, there's a lot of negative feedback mechanism. So this is where the FSH threshold concept comes, where there's a peak rise in FSH for selection and dominance. Once there is a dominance, there is a negative feedback by the estrogen, which reduces the FSH, and this causes atresia of all the other follicles, and thus this exhibits a monofollicular development, which is very crucial for the human reproduction. And over here, what we see is mainly the once there is a monofollicular growth, the follicular, all the other follicles go into atresia, and post ovulation there is formation of the corpus luteum. Now this is a where we have depicted that, as I told you, that AMH is a very important, which reflects the ovarian follicular pool, and this only reflects the dynamic egg reserve. There is also a pre-established reserve, which is a primordial follicle, which does not get depicted by the AMH. So once there is all these regulators which control the ovulation, it is not just FSH, LH, but there's a lot of paracrine regulators like insulin, IGF-1. So these are all equally important for the follicular growth. And once there is ovulation, the follicular depletion happens. As the follicular depletion happens, this AMH again has a feedback mechanism for the BMP, activin, and the KIT ligand, which will push the follicles from the primordial full back into the dynamic reserve. So this is a balance that is maintained in the oogenesis process. Now coming to the what happens during ovulation. Now when the primary oocyte divides, its nucleus, which is called the germinal vesicle, it breaks down, and the metaphase spindle migrates to the periphery of the cell. Now this is a very important difference to the spermatogenesis process, because here what is happening is that by the end of telophase, one of the two daughter cells is going to take the entire cytoplasm, and the other is left only with the nucleus. So this is a reduction division. So the smaller cell is called the first polar body, and the larger cell is referred to as the secondary oocyte, which becomes the haploid. Now, during the second division of meiosis, a similar unequal cytokinesis takes place, and most of the cytoplasm is retained by the mature egg or the ovum, and a second polar body receives little more than a haploid nucleus. Thus, the oogenic meiosis conserves the volume of oocyte cytoplasm in a single cell rather than splitting it equally among the four progeny. Now, all these are very important, because when we talk about oocyte competence, it is not just important to reach the particular follicular size. So whenever we take use of all these, this is very important for us for timing of the trigger. Whenever we plan patients either for any ART cycle, whether IUI or IVF, we have to ensure that by the time the oocyte reaches for fertilization, there should be a very good competent oocyte with respect to nuclear and cytoplasmic maturation. And today we are also talking about epigenetic maturation. So this is where we see the ultrasound picture of the scan, and where we see the antral follicle, that is small antral follicles. After stimulation, they reach to the large antral, the 10 mm, and finally the pre-ovulatory follicle. So why all these are important is mainly with respect to the trigger. As I said, for IU and IVF, the standard thing what we do is between 15 to 20 mm for IUI, and between 15 to 22 mm follicles are triggered at the time for IVF. And usually the egg retrieval is timed for about 36 hours post the shot, 35 to 36 hours. Now, coming to the results, now how do we maintain results is by effective triggering. So what does the evidence say for triggering and timing on IUI? Because see, anything about oogenesis, anything about oocyte competence, why we have all this knowledge is mainly because to ensure that the right timing is where the oocyte is coming out of the follicle. And what they've seen is, even if you compare to doing an IUI where there is no trigger shot, the success rates are 5% versus giving a trigger shot increases the success rates to nearly 18%. And in fact, what they tell is the later the trigger, it is better. There are studies where they're given the shot even after the IUI, because that's when the oocyte, the moment ovulation happens, the oocyte death also starts. So giving a shot after the IUI also improves the pregnancy rates to nearly 19%. In fact, the later the IUI, that is IUI 42 hours after HCG injection, significantly increases chances of fertility versus at 36. You can see the pregnancy rates. By doing an IUI at 42 hours after HCG, we have 28% versus at 36 hours, 14%. So nearly doubled. So there's always a significant effect of delaying the IUI till nearly 48 hours after HCG injection, where again you can see the pregnancy rates of increasing, nearly doubling, where you prolong the IUI to 48 hours instead of 36 hours. Now coming to IVF, if you look at the IVF success rates, which have improved from last 4 decades, from 5% to nearly more than 50%, among all the advances that have happened, the main thing I think we should attribute it to the super ovulation and the controlled ovarian stimulation. It is only because of that we have reached these high success rates with IVF. So from ovulation induction to super ovulation to COS. So the main thing about COS, where we know the role of oogenesis, the knowledge of it helps is mainly by preventing the premature LH surge, controlling the time of ovulation, and to time the insemination during IVF or ICSI. So this multifollicular development is mainly achieved by increasing the dose of the gonadotropin or increasing the threshold of exposure. So that is how we prevent the negative feedback mechanism and try to recruit. Instead of this atresia of all the follicles, we are promoting the development of all the follicles to reach the appropriate window for trigger. So there are a variety of COH protocols that we use with all the mixed success. So I would not tell one singular protocol works to get the best oocyte quality. It should always be individualized to each patient. And this whenever we individualize stimulation, what we should look at is mainly we should reduce the risk of OHSS. At the same time, we should reduce the risk of cancellation because of a poor response, and try to get an appropriate number of oocytes for maximizing our success rates. Yes, there are a lot of markers which indicate the stimulation strategies, but today with along your hormonal markers and functional markers, genetic markers are coming in which could have a major role in the future, which is called the pharmacogenomics of individualized controlled ovarian stimulation. Now, just to go a little about the IVF cycles, and you want to know regarding the oocyte coil. It's like we are trying to tell the person who's living inside by looking at the house of the person. So that's very difficult, but that is what we have when we time IVF cycles and plan the best trigger. So we all depend upon the follicular tracking, follicular size. So now we want to know the correlation, what is the optimal size of the ovarian follicles during oocyte collection. So if you look at it here, the key word is at oocyte collection. See, when you look at a study, many talk about the optimal size during the trigger. So this is where they have done the follicular size at the time of collection, where they have told that 16 to 27.5 mm diameter had the maximum blastocyst formation per follicle. And if you look at the day of trigger, the studies have shown that it is between follicles between 12 to 19 mm on the day of trigger, which contributed the most number of mature oocytes that you get. Now, if you look at the type of triggers, whether you give a HCG trigger, whether you give an agonist trigger, or whether you give a dual trigger, will that change, your decision? But here, what the study told is the triggering mode did not influence the oocyte recovery rate. In fact, you got better oocytes when you better mature oocytes when the follicle sizes were more than 13 mm. But irrespective after that, after 13 mm, whether the oocyte came from a 14 mm or a 22 mm follicle, as long as you got mature oocytes, they had similar fertilization, top quality embryos, and the live birth rate. So here, now what they confirm, what they are talking about is, how do you talk about oocyte competence? Will you look only at the follicular size, or would you look at the morphology of the follicle, or will you look at the perifollicular blood flow? So here, what they saw was that as the follicular size went on increasing, the Dopplers also will start changing. You will have a peak systolic velocity of the follicle, which will fall, and the RI falls. So all these are indicative of a better mature oocyte within that growing follicle. Coming to the ovulation trigger and the OPU time interval, because this is something very standard we do between 35 to 36 hours. So when they studied that, will the results change when you extend the timing of that interval? Now, this was compared with respect to the kind of protocols that you were doing, whether long agonist protocol, short agonist, mild stimulation, and GnRH antag. So here, what they told is, whenever you are long agonist protocol, you have got a very long time interval between 35 to 40 hours. Also, you can time your OPU. But when it came to mild stimulation and GnRH antag, you have a very narrow window of timing or retrieval between 34.5 to 36 hours is the ideal time. So does expanding the time help? Now, there are a lot of studies which tell you that the best time for insemination or injection is between 38 to 39 hours, post retrieval from your timing. And usually the authors tell that more than 41 hours has a negative influence on your embryological and clinical pregnancy outcome. But if you look at this study, which is recently published in 2021, it's from more than nearly 9,000 ICSI cycles, where they have compared between 7 categories, where they have seen between less than 36 hours, the time for ICSI, that's the injection, 36 hours, 37, 38, 39, 40, and more than 41 hours. Here, even when the ICSI was done more than 41 hours, post-triggering, there was no difference between your maturation, fertilization rate, or your blastocyst, or the embryo utilization rate also. And similarly, when they compared less than 36 hours injection to 38-hour injection, again, there was no significant difference. The only key word that you have to remember over here is do not denude the oocytes, that's all. The denudation was performed immediately prior to the injection. So as long as that is the case, even more than 41 hours does give a good outcome. Now, is it follicular size or follicular volume that you need to worry about? Now, here they've done a size between because see, these are all clinical things that we take decisions upon to actually retrieve the oocytes to give it to the embryologist. So if you look at the follicular volume, they tell it is best between 1 and 6 ml. But even in less than 1 ml follicle, if you have got an oocyte which is mature, again, the results are the same. It doesn't difference, because finally it comes to the grading of the oocytes. The better oocyte quality is what determines your better pregnancy rates. The follicular size, even if it is 13, 14 mm and you're getting mature oocytes out of them, the results don't matter. So follicular size only should be used for probably the time, the trigger at that right time. Now, why do we understand so much about oogenesis is because now there are so many studies coming on IVM also, where you realize that yes, it is not just IVF, but even in IVM, where you are actually you are not giving much of a stimulation to these ovaries, and you're collecting the follicles from the small and medium sized antral follicles, and meiotically you're maturing them in vitro from the GV to the M2 stage, and even these are giving quite good results. But yes, it's a long time to go to master this art. As you develop, as we see IVM giving results, the question comes that could we get into oogenesis from the beginning? That means even from an earlier phase than what we're doing in IVM. That is called the germ cell renewal by new oogenesis, which has been proposed as a possible mechanism to replenish the pool. However, again, it is disputed regarding the studies. So this is what they tell, that in vitro development. Now, where you go back and see whether you can even develop from the 4th month stage of development from the primordial follicle to the secondary follicle. So understanding oogenesis better probably will give us more insights into delving deeper and deeper into this, and probably helping patients with POI. And because I think we have almost conquered IVF, I would say, trying to master the IVF. And this is what probably is newer thing is the new oogenesis. And thank you. Yeah.
Thank you, Dr. Vandana. That was an excellent talk. And briefly, you summarized all the things. I just wanted to bring, sort of focus on one fact, that we all talk about egg quality and the, reserve eggs and everything. But what we are forgetting is that lifestyle modification is the key to getting a better egg quality. Because all the patients, at least now I am seeing, I don't know, but there is a very sharp decline in the quality as well as the number of eggs recently. The patients who are coming almost all have a little, not so good lifestyle, with the lifestyle disorders, with low ovarian reserve. As young as 30, we are seeing low ovarian reserve. So this is very important, that we should also focus on giving them, imparting knowledge to them about the lifestyle modifications, working on their body, improving their lifestyle. Having a healthy egg starts from having a good night's sleep and good food. That's what I think is more important, spreading knowledge about this. Other than that, it was a wonderful presentation, and our all basics are, cleared up again. That's it from my side. Thank you so much. It was great listening from you.
Lifestyle is definitely good if you could change here, but then the problem here is the environment cannot be changed. There's a lot of environmental damage that is happening. The external environment, which probably is one of the factors, that you see the ovarian reserve depleting over time, which is modifying the genetics. The environment modifying the genetics is what I think is the main culprit over here. Yeah,
I agree, Dr. Vandana, but even the environment also. So, like, for example, I believe that if we are at least taking care of, to some extent, our lifestyle, like the food we are taking, daily habits, eating habits. If we go back to our roots and start eating the combinations and all which we were using at those times, to some extent, we will be able to overcome that.
Yeah, definitely, yes.
Thank you so much, ma'am, for the wonderful presentation. I would request you to stop the sharing your screen, ma'am. Vanda, ma'am.
Yeah, done.
Ananta, can you take over for the questions?
Yes.
No, wait. Any questions are there?
From the audience.
Ananta?
No, sir.
No, right?
No, sir.
In that case, I have one question for madam. Ma'am, this is for the younger audience, like who are attending the talk, especially the embryologist, younger embryologist. So, can you tell us what do you prefer with respect to trigger, agonist or HCG or combined?
I usually prefer a dual trigger because it is more physiological when you add, and at the same time, we have to acknowledge that HCG also has a better role. So sticking only with the agonist trigger is not what, even if we are freezing all, it's not just because we are freezing all that we want to give agonist trigger. We would like to give a dual trigger wherever there is no risk of OHSS, because I feel that we do get a better, oocyte, the maturity level, everything is better. That's our practice actually. Yeah, wherever possible, we give a dual trigger.
Right.
We usually give a dual trigger only, and we reserve the agonist trigger to only cases where we suspect OHSS. Otherwise, we give HCG trigger only. Dr. Vandana, like what combination are you giving in dual trigger? Like what are you giving? HCG plus DECA, and at what time interval?
No, we are giving both together.
Okay, both together.
Yeah,
so this is like 0.25 orbital plus 0.1 milligram DECA?
Yes, depending on the weight, sometimes we do give 0.2 mg also. Yeah.
Okay. And have you you showed a beautiful study about the timing and interval and everything. Have you ever seen that some centers I see that they are doing ovum pickup at 35 hours instead of 35.5 or something? In our center, we prefer to do at 35.5. So is there less number of M2s retrieved in those cases, or you give a longer duration of before denuding the eggs? That's it?
No, actually, we usually time it for 35 hours, but again, invariably, it does happen by, we keep that lag time of 30 minutes. Again, it happens by 35.5 hours.
And we also do it 35.5.
But yes, some patients, sometimes we do time at 36 hours. Or somebody who's on the, higher age, like, above 39, above 40 years, we do time them sometimes at 34 hours also, depending on the follicular size. If the follicular size has gone, slightly larger, more than 20, 21, and she's higher age, because we don't want that, sudden rupture before the retrieval. Sometimes we time 34, yeah.
And in these cases, do you trigger also a little early, not waiting till 19, 20, maybe at 16,
exactly.
17?
Yeah, that is why we trigger. That is the reason we need to think of the, the size at the time of pickup sometimes. That's why 18, 19, sometimes we just trigger them. Yeah.
Yes. So in what I do a lot of natural cycle where we are retrieving single follicles. So I just wanted to put forward that point also, that natural cycle transfer per transfer, the success rate is much higher. But per pickup, usually it drops down, because sometimes you don't get a good egg or don't get a good embryo. You have to cancel the cycles and postpone, and those things happen. It was wonderful hearing you, listening to you. It was wonderful. Thank you so much.
Thank you, Dr. Nisha. Thank you so much. Thank you.
Just to extend what Dr. Vandana madam has said, Nisha ma'am, see the thing is for us being an embryologist, we count from the time of pickup.
Yes.
When renewed. So of course, if it is like 35.5 or 35 or 34, we calculate from the pickup time when to denude them. So it is like almost standardized, like standard of care, I can say.
Okay. But have you ever tried keeping the, if you get you think that the eggs are a little smaller, maybe you can make it out for after the pickup. Do you try to keep it prolonged incubation without denuding? Do you do that?
Yes. Thanks for the technology. Best optics we have, and thanks to Dr. Vandana madam for giving us best optics. That's what we do, especially when when we retrieve less number of COCs. We partially denude them and we keep them, we observe them under the microscope. We see whether there's GV or M1 or M2.
Okay.
And that's what we're practicing. If it is partially we denude and we see them, based on that, we're taking a decision whether to keep for IVF or ICSI.
Oh, that's
or whether to wait for some more time for denude for the proceeding for the ICSI.
Okay. And when we talk about this, I have one more question, please, because this I was in my mind since long. So I heard somewhere about incubating the eggs with some sort of HMG in it. Have you ever tried it?
Not
Some
really.
gonadotropin added to the culture media?
No,
Have you
no,
ever tried it?
not really. The thing is, ma'am, the media what we provide is highly optimized. Like say,
Okay.
whatever the culture media, it does have some promoting growth factors which helps for maturation of the oocyte. And moreover, like if it is not matured like 3 to 4 hours post pickup, then chances of like maturing after that will be very
Less.
less.
Okay. Okay. Got it.
Very tempting, no, Dr. Nisha, as clinicians, just to put a HMG over there.
Yes.
I have
Get
asked this
into-
question actually to my embryologist, Dr. Akash.
Yeah.
Put a little HMG, you know.
Actually, it was a wonderful talk, Dr. Vanna, and nice discussion. I would like to jump in over here. And I would say, yes, it's very tempting, but we'll see during the course of the next talk also that, many a times just removing from the inhibitory factor, which the follicle keeps, will allow your oocyte to mature. But what it has been shown is, if it is happens, like said already by Dr. Sanket, like last couple of hours, if it's within 2 to 3 hours, if it's happening, it's just maybe the slight biological variation what you're seeing, or maybe many other things over here. But if something does not mature within the first 6 hours, 4 to 6 hours, that means it was inherently there is some defect over there. And these type of oocytes, though they may go to M2 as such, they may fertilize as well, but the chances of forming of blastocyst and giving a healthy pregnancy is quite less actually with this. So if something does not mature within 2 to 4 hours, we may improve our fertilization rates, we may see a better cleavage stage embryo, but then the chances of pregnancy would be quite low.
I understand. I understand. But sometimes it is better to tell to the patient, no, that yes, we have something.
Yeah. I mean, I said there are like many benefits, which
Yeah,
include our indicators as well, which would be improving. But then if those are the oocytes, then definitely we'll be going ahead. And as such, we also do keep them for what we say the emergency IVM kind of thing, not the proper IVM. And we do use them if it forms, and preferably we keep them till the blastocyst stage and then use them.
Rescue IVM.
Yeah, rescue IVM.
Great talk, Dr. Vanna. Thank you so much for an excellent brief talk.
Thank you for the opportunity. Thank you to all of you. Thank you to iHera. Yeah, thank you. Bye.
Bye, ma'am. Thank you so much, ma'am, for giving that clinical perspective of the beginning of the largest human cell, that is the oocyte. And yeah, it's time for the poll. Let's take some few questions. Like Ananta, you're ready with the poll?
Yes.
Yeah, please go ahead.
Can we have the questions?
Yes, ma'am.
This is for audience.
Yeah, small statement.
question.
Who
Yeah,
has
first question.
listened to Dr. Vandana's madam talk? It will be very easy for you guys to answer these questions.
So, shall we start the poll question? The first question is, primordial germ cells undergo dash cell division and form the primary oocyte. The options are mitosis, meiosis 1, meiosis 2, both mitosis and meiosis.
Audience will get around 15 seconds per question.
So the second question is, diameter of the normal mature oocyte is greater than 300 micrometer, greater than 200 micrometer, greater than 120 micrometer, it will be less than 100 micrometer.
A screen is not seen.
Question you are not able to see?
Yeah. Yes.
No, Shita, they are visible.
You need to answer on your chat. The questions are there on the chat.
Actually, the polling, they will be choosing the options, right? They no need to answer through the chat. They can click the options.
Yeah, directly on the screen they can click.
Yeah. The options for clicking will be available only for the delegates.
Delegates, not for the
not for the faculties and
Okay.
Jagananda, please go ahead with the third question.
Third question is, the average diameter of a mature Graafian follicle is dash. So the options will be 20 centimeter, 2 centimeter, 20 millimeter, both B and C. Average diameter of a mature graphene follicle is 20 centimeter, 2 centimeter, 20 millimeter, option B and C, both B and C. I think this is one of the question which we got in yesterday exam.
Yeah, 2021.
Yes, sir. Shall we move on to the fourth question? So
Please.
till puberty, oocyte development get arrested in dash. The options will be prophase. The option 2 will be metaphase 2. Option 3 will be metaphase 1. And option 4 is anaphase. Option 1 is prophase. Option 2 is metaphase 2. Option 3 is metaphase 1. Option 4 is anaphase. We can move on to the fifth question. The fifth question is mature metaphase 2 oocyte, that is mature M2 oocyte, has dash chromosomes. Now options are 23 chromosomes. Second option will be 23 pairs of chromosomes. Option 3, that is third option will be diploid set. And the fourth one is 46 chromosomes. Fifth question is mature M2 oocyte has dash chromosomes. Options are 23. Second option is 23 pairs. Third option is diploid set. And the fourth one is 46 chromosomes.
Well, that's the end of the first 5 questions of the poll. Can we have the results, please?
So the
Please go ahead.
audience answered the first question. The right answer will be mitosis. Primordial germ cells undergo mitosis division to form primary oocyte. So the correct option is mitosis. So 60% of the audience have answered it correctly. Coming to the second question, the diameter of the normal mature oocyte is, it should be more than 120 micrometer. So the 75% of the audience have answered it correctly. Average diameter of a mature follicle is, graphene follicle is, the answer is 20 mm. It is correct only, but 20 millimeter and 2 centimeter almost it is same, right? So both B and C is the right option. Only 33% have got it right. And fourth question, till puberty, oocyte development get arrested in? Till puberty. So onset of puberty only, the oocyte starts to get mature. So the correct answer is prophase. And the fifth question, the right answer, mature M2 oocyte has dash number of chromosome. It is usually haploid. So it is 23 as the right answer. Only 33% of the people got it right. So can we move on to the second session of this day?
Yeah. Thank you so much, Ananta, for that questions. And thank you, audience, for the participating actively in this poll. And yeah, without further delay, I would like to introduce the next moderator, Dr. Vijay Mangolisa. Can we have the CV, please? Technical team, can we have the CV of Dr. Vijay Mangoli, please?
It's okay, Sanket. We can just go ahead, no problem.
Oh, no, sir. I mean, at least you have to introduce the speaker, sir. So, Mr. Vivek?
sir?
Can you have the CVs, please?
Yeah.
Yeah,
Okay. I'll share my screen.
sure.
Uh,
Basically, the series is designed in such a way that, like, in this particular webinar, we'll be talking about only about until metaphase 2 oocyte. The next followed up webinar will be dealing about later parts. Like, so, well, we have Dr. Vijay Mangaleshwar with us, and he's a PhD and a laboratory director at Fertility Clinic and IVF Center, Mumbai. He's been associated with Human IVF since 1986, and he's a Secretary General of Indian Society of ISAR from 2009 to 11, and he's a chairperson of Embryology ISAR from 2014 to 16. And he has published numerous publications in very high indexed and peer-reviewed journals, and has also sir has presented a lot of presentations in national and international conferences, and till date, he has been contributed more than 49 chapters, like almost 50 chapters in medical reference books, and he's a recipient of Lifetime Achievement Award in 2017. Sir, over to you. I'd like to request you to introduce the next speaker.
Thank you, Sangeet. Thank you very much. And I really appreciate your introduction. And let me introduce Dr. Akash Agarwal. Dr. Akash Agarwal, he is an MBBS turned HRA certified clinical embryologist. This is really a good combination to become an embryologist, I must say. He's alumnus of the Osmania Medical College and practicing male fertility and embryology since 2011. He is a topper in HRA clinical embryology in 2019. And he's actively involved in academics and co-authored many book chapters. And I must say that I have listened to him and I have read his articles also. A very thoroughly researched articles, and he writes. And although he has a keen interest in the male factor particularly, but today he is going to enlighten us the basics of some of the oogenesis. So over to you, Akash.
Thank you, Vijay sir, for the kind introduction. So after a very enlightening talk on the oogenesis, I would like to carry forward from over here in terms of the plays and pauses. What are the checkpoints which are present in a oocyte as such? Just give me a minute. I'll start sharing my screen. Is it visible, Dr. Singh?
Not yet.
No, sir.
I've started to share already.
No, sir, it's not visible.
Try to redo it again.
Let me just stop and once again. Is it visible now?
Yes, sir.
And changing of the slide.
Yeah.
Okay. So we have already heard in the previous talk to a quick recap. So we usually see for the Graafian follicle to be mature, in which we are supposed to have a meiotically competent oocyte which is arrested at the prophase 1. And usually you get some external cues should be present for the resumption of this meiosis 1, which usually is the hormonal cue and which is usually the LH surge which happens just before the ovulation. Or alternatively, it could be the inhibitory factors which are present in the environment of the follicle. So once you take out them from the in vivo to the in vitro, automatically the inhibitory factors are removed and it would automatically spontaneously progress for the meiosis resumption. So in COH, your stimulation cycles, usually we use HCG as a surrogate for LH. Normally, we see metaphase to be progressed and ovulation to occur somewhere around 36 hours, at which it would be a metaphase 2 arrested oocyte as such. And usually we are going to see so many steps over here which are involved in the maturation phase as such and the meiosis and so many checkpoints, but we rarely see a failure as such of all the oocytes to mature. See, 1 or 2 oocytes to mature, that could be because a oocyte has started to develop later, could be something to do with the stimulation or the patient profile as such, but all of them getting arrested, it's a very rare thing, and there are very, very few cases which have described such. So when can they get arrested? They could get arrested during any part of the cell cycle. So this is just a quick recap of what majority of us knows, your cell cycle consists of your G1 growth phase and the G2, which is interspersed with the replication phase, the S phase. And post G2, you have the M phase, which is nothing but the mitotic phase, which includes your nucleokinesis as well as the cytokinesis, where the nucleus divides first, followed by the cytoplasm in the regular order. And it has, each one of the phases has its own importance. I'm not going to go in detail of that. Basically, G1 is your growth phase. Your synthesis of your organelles will be there, and macromolecules and the energy substrates as such, they are going to be accumulated during this phase. From here, when the cell gets a signal externally, it goes into this phase, the synthetic phase, where the DNA replication happens. Along with the DNA replication, centrosome replication also happens hand in hand. And post this, the cell enters another phase of quiescence relatively, which is another G2 phase, at which other cell cycles, checkpoints and others would act, and further accumulation of proteins and organelles will happen. Post this, the cell enters your division phase, which is nuclear division, followed by the cytoplasm division usually. So at this point of time, the thing we need to think is, where are these checkpoints present usually? So at which point in the cell cycle would these checkpoints be present? So the checkpoints are present at multiple points in the cell cycle as such, but majorly we take as 3 checkpoints. The first one is your G1 checkpoint, which is usually called as the restriction checkpoint in mammals, or which is called as the start checkpoint in the yeast, because this is where the cell, in this case the oocyte, gets a signal that it has to enter the meiosis. Otherwise, it would remain in the same mitotic stage as such. Next is your G2 and M interface checkpoint, where usually between the G2 post the replication, the cell has to enter the active cell division as such, nuclear division. So at this time, there would be a checkpoint which checks for whether the duplication has happened properly, once and only once it has been duplicated DNA, and there's no DNA damage as such. And the last important checkpoint would be the metaphase to anaphase transition, which is also known as the spindle checkpoint or spindle anaphase complex, SAC, by various names it is known as. And this is considered to be the most important checkpoint and the last major checkpoint. So these are the checkpoints which are present in mitosis. But the thing to be known is, these checkpoints are present in meiosis as well. Majority of the checkpoints which are present in mitosis are present in the meiosis as well. So these are the various checkpoints that we have been talking about: the G1 checkpoint, the G2 checkpoint, and the spindle assembly checkpoint, and the factors which usually control. Dr. Vanna was talking sometime earlier about how important is the cell size, the oocyte size as such. So we see in the G1 first checkpoint itself, the first determinant would be the cell size, growth factors, and the damage, whether anything is present in the DNA or not. And if there is any of these conditions are not fulfilled, then the cell cycle checkpoints ensure that the cell does not carry forward in your cell cycle as such, and it remains arrested. It could be a G0 phase. If the damage is little, it can get corrected and again the cell re-enters the cell cycle, or it could be a permanent stage as such, and it could be even a stage where it could undergo apoptosis as well. So these are the common cyclins that are usually involved in these checkpoints, major checkpoints. There are many cyclins. I've just highlighted the ones which are the important ones. Usually it's the cyclin D and CDK4, which is involved in the G1 checkpoint, which checks for the DNA damage. It ensures that the cell enters and there is no much of DNA damage. And similarly, you have at the time the cell enters into the S phase, the cyclin E and CDK2. And similarly, for during the replication phase, you have A2. The short form is like cyclin A and CDK2 is called as cyclin A2 as such. And during the mitotic phase entry, it's usually cyclin B1. These are the usual common checkpoints which are involved. And this is just one slide I thought I'll put, like each cyclin can be involved at multiple places, and each cyclin and CDK complex has multiple activators and inhibitors, and there is a huge complex interplay which is going on over here, that which results in a proper development of the cell cycle. So we have heard the word cyclins. Cyclins will be hearing commonly. So what are these cyclins and why is the name called as cyclins? So usually these cyclins are the proteins which will accumulate during the interphase, and usually they are degraded rapidly at the end of each mitosis. And the discovery is so important that the discoverer of cyclins, Tim Hunt, was awarded a Nobel Prize in Physiology in medicine for this, along with Morgan and Hartwell as well. So direct evidence for these cyclins was provided in 1986 by Ruderman, when she had shown that the microinjection of the cyclin A into the frog oocytes results into the direct transition from G2 to M without the hormonal transition, hormonal stimulation, which is required normally. So this is one pictorial depiction again of how a restriction point checkpoint would be overcome. You could have growth factors which could be in response to multiple stimulus as such, and this results in a synthesis of the D-type cyclins. The point to be known here is usually your CDC subunit of this cyclin and CDC units will be present perennially, will be present continuously throughout, but it is the cyclin level which will be increasing and decreasing during the cell cycle. That's why the name cyclin as such. So these growth factors result in the stimulation of the D-type cyclin, thereby it helps in overcoming the initial part of it. Then one of the example of how a DNA damage can result in a cell cycle checkpoint, where usually if DNA damage happens, there'll be a concomitant rise of the P53 levels. This P53 gene upregulation will in turn cause the rise of P21 mRNA more to be produced, transcription more of P21 gene. And this P21 gene will in turn result in the changes of the CDKs, cyclin-dependent kinases, and the PCNA, which directly inhibits the DNA replication as such. So this is just one part of it. And another part of how your CDC25, how your checkpoints during the replications happen. Suppose there is a DNA damage or the replication is incomplete. If you can see towards the right side of it, usually there will be checkpoint proteins called as CHK1. So these will be associated with the DNA. If they are associated with the DNA, the CHK1 will be free to inactivate the CDC25. So this CDC25, as you can see towards the left side of the image, usually results in formation of active MPF. So if CDC25 is not available, then automatically there is the MPF, which is maturation factor, will not be available actively, and there happens a G2 arrest as such. So entry into meiosis. So we have been speaking about all during the mitosis during this time, and all these factors apply to meiosis as well in the mammalian oocytes. So in addition to this, what are the changes that we see? See, one thing we need to know is that meiosis itself is quite different from mitosis. And we see that it is dimorphogenic in that the female and male mammals, it is quite different. In females, it develops during the fetal part itself, where the germ cells will enter into prophase 1, and there they are undergo a long period of arrest for decades till they resume their division and may be ovulated or may undergo apoptosis. In contrast, usually for the males, it usually happens post-pubertal when you can see waves of this cell division happening. So when we speak about oocyte maturation is important. So what exactly is oocyte maturation? So oocyte maturation usually involves a series of changes both in the nucleus as well as the cytoplasmic component of the oocytes, which makes it capable of achieving fertilization as well as supporting a normal embryonic growth post-fertilization. So it involves both nuclear and cytoplasmic. Nuclear, something which we can see easily, which is extrusion of the second polar body as such, the metaphase 2 oocyte. And the cytoplasmic, which we don't see many of the times. It includes accumulation of proteins, changes in the mitochondrial number, as well as the location where they are localized, Golgi apparatus, and the other organelles. So your G2 phase checkpoints usually operate during the meiosis as well. One point to be noted over here is usually the G2 phase checkpoints are much more active during mitosis, but while as far as meiosis is concerned, usually they are more active during the initial part of the G2, the early phase. But once the cell enters the late part, these controls are less effective. So this is one of the reasons they say maybe human reproduction is not that much effective, probably because of this lack of a proper G2 phase checkpoint. And once this G2 phase checkpoint is overcome, usually which is in response to a luteinizing hormone surge, LH surge, we see that there is a resumption of meiosis 1 and the extrusion of the first polar body and the oocyte to the rest in the meiosis 2. So what are the special changes that we see in terms of meiosis when we say the checkpoints in terms? What are the things that the cells has to overcome specifically? The first thing is the prophase. Usually your cell cycle in some prokaryotes or E. coli or yeast is as short as 20 to 30 minutes, the entire cell cycle. Whereas in mammals, the average length, if you can take it, would be something around 23 hours. But over here, you have a cell which is arrested in fetal stage and would undergo resumption at the adult puberty stage. So there's a long prophase which is happening. There's a recombination, which is a new thing which does not happen in the mitosis. Next would be there should be an absence of S phase. See, usually when we saw the cell cycle diagram, we could see that there's always a G1 followed by an S and then a G2 phase, and then the division phase. But over in meiosis, this replication would not be there. This S phase would not happen as such. So that is another challenge, that the S phase should not happen. And how the chromosomes will be separated. See, it's a reduction division as such, your meiosis one, so the number of chromosomes will be separated. Mitosis, there's separation of the sister chromatids, but over in meiosis, there's separation of the homologous chromosomes. So how is this achieved? So the cell will respond to this each challenging situation in a different way. It is so different that you are replicating cells within the germ cells versus the testicular replicating somatic cells. There's 1268 loci. One of the study found that there are differences in terms of activation. So probably there's a lot which is going on inside a cell which we don't know as of yet, and how exactly meiosis is regulated. At the meiotic entry checkpoint, it's once again the same CDK cyclin kinase activity which will regulate it. And anecdotally, this was the first factor which was as such found. At that time, it was not properly understood what is the factor, and it was called as maturation promoting factor, because if you would extract the cytoplasmic extracts from the mature oocytes and you put them inside, inject them inside the xenopus, the frog as such, which are immature, it will induce maturation. So that means it's not the nucleus, but the factors which hold for inhibition and which helps for progression of the cell cycle are present in the cytoplasm. This was demonstrated in around 1980s, late 1980s. And this MPF, maturation factor, was found to be nothing but a heterodimer, which we call it as the CDC2 cyclin B heterodimer. And this is present throughout. It is just an example of how complicated things can be in terms of a simple regulation. In terms of CDC activation, they say CDC2 and cyclin B dimer is present continuously heterodimer, but it undergoes phosphorylation. If you see the top left of the diagram, where your various enzymes CDC2, V1 and MIT1 will undergo the phosphorylation, and this phosphorylates this CDC not only at the threonine 161 place, but even at the tyrosine for the 14 and 15. So your threonine at 161 is important for the activity, but this additional phosphorylations will result in the inactivity of this CDC2 cyclin B complex as such. So again, this needs to be dysphosphorylated, which is done usually by CDC25 phosphatases. So activity of CDC25 phosphatase was remove the phosphoryl group at the 14 and the 15 position, thereby ensuring that the mitosis and meiosis is being entered into. So this is again a depiction of what all changes will happen, and how, and each of this step can be regulated one or multiple transcription factors as such, and each of the factor is again related and again controlled by a multiple converging pathways. So whenever there is a CDC25B is lacking is there, obviously the cell would not go ahead in the meiosis, and you can see cells which remain in the germinal vesicle stage as such. So there are many other factors which are over here which can overcome this downstream products as such of this. One of the important one being the Ringo. I'm not going to go through all the list of this since it's a quite long list and time may not be sufficient. Other than those, what is during the prophase one that we need to see over here? One thing is additionally the synapses and the recombination, which normally would not happen during the mitotic phase. So this is the additional checkpoint where it is required. So usually if your CDK2 is inactive, it will result in the improper distribution of the SIP protein as such, and synapses would not be proper. And again, synapses and recombination are commonly linked together. And if they don't happen properly, then there'll be a DNA damage. And for this, you have additional DNA damage checkpoints, which are RAD1, ATR, TOP1, BRCA, and CHK1, one of the examples which you have seen earlier. So all these proteins help as a damage checkpoint proteins. These are present in mitosis as well as meiosis as well. So this is how the different proteins for these checkpoints work. Recombination checkpoint, RAD50, DSB, the red one, which is again dependent on red one or independent of red two, and something called as the synapses checkpoint, which is totally independent of this all, and it is dependent on the PCH2. So this is a pictorial depiction of how and where the checkpoints would be appearing. And the initial part would be the left side of the image, where you can see the CDK1 and the cyclin are the primary controllers as such, whether the cell enters further into the meiosis one or not, and it undergoes the germinal vesicle breakdown. So next part would be the absence of S phase. Like I said, it has to be tightly controlled that there is no replication of the DNA happening between meiosis 1 and meiosis 2. So for this, what you require is the CDC2 cyclin B activity is there. So it goes down to a certain level, but does not reach the total lowest level. In that case, this activity is continued to maintain. So what happens with this continuing activities, G1 and G2 may happen transiently as such, but the cell division, DNA synthesis does not happen. So that is how the cell overcomes this additional challenge of not having a replication between meiosis 1 and 2. So this is where this would be present. And usually it is helped by even having some lower levels of APCC, which we'll see later. Now the last part would be the anaphase promoting complex. It is considered one of the primary checkpoints in mitosis as well as in meiosis. It usually involves the formation of the spindle and the correct attachment of all your fibers, the microtubule fibers originating from the centriole, which will attach to the kinetochores, the central part of the chromosome. And then when the tension is correct, when all have been attached, it results in a separation, starting of the anaphase. One thing which I found surprising while I was preparing for this, I came to know that probably the meiosis is acentriolar in mammals. So it does not involve the presence of centriole as such. Centrosome is present, but in some mammals and many mammals, it's acentriolar. Centriole is absent during oogenesis as such. It is present for the sperm, but not for the oocytes. So this anaphase promoting complex, APCC as we call it, it is nothing but a ubiquitin-based ligase complex, and it involves the activation of the proteasome, where a part of it is separated, ligated once it receives the right signals, and results in the entry into the anaphase. So it would be present somewhere in the middle part of it when the APCC levels would drop down between the meiosis 1 and 2. And there are certain things which need to be additionally taken care. It needs to be tightly controlled, the entire junction, how it is being done, the levels where it increases, decreases. And again, they needs to be modulated during the chromosomal segregation. Again, it involves separately, because where and when it would act makes a huge difference in meiosis and mitosis, because in meiosis, there should be separation of homologous chromosomes as such. But whereas in the mitosis, there need to be separation of the sister chromatids present on attached to the same kinetochore as such. And lastly, during the metaphase 2 arrest. So this usually delays are inherent to it. It thereby indicates that the delay in metaphase 1 is not just because of some arrest factor, but even because how complicated it is. Because usually during your mitosis, what you require is the sister kinetochores to be attached to the two opposite poles of the spindle. One would be present here, one sister chromatid, and the other spindle to the other pole would be attached this other sister chromatid, the kinetochores at that point. And once they have all have been arranged along the metaphase plate, they would be separated by the tension which is generated by this activity of the APCC. So over here in meiosis, what you require is not just that the sister kinetochores of each chromosome should be attached to the same pole, which is the co-orientation as we call it, but the homologous chromosomes which are present should be attached to the different poles so that the correct reduction division happens. So if you have two copies of, say, chromosome 10, one of the chromosome 10 which has divided should be attached with both the sister chromatids to one pole, and the other which has come from the other parent should be attached to the other pole. So this is called as biorientation of the homologous chromosomes and co-orientation of the sister kinetochores. So this is much more complex than what is present in mitosis, and the exact mechanism by which this happens and how the cell controls this is not still very clear over here. And how does the APCC complex works as such? Usually it has something called as securins which are in the inactive stage, and once these are activated by the factors which regulate it, you'll see that there is a separation. Separases are activated, and these separases will cleave the cohesin molecules which are present in between the sister chromatids and even at the kinetochores in mitosis, and they'll cleave only separate during the mitosis as such, resulting in the separation of the sister chromatids to the opposite pole. And this securins and separases even work similarly during the meiosis 2 as such, whereas during meiosis 1, again their action differs because the distribution of the recate protein which is present will be present during the meiosis 1 only along the arms of the chromosome as such. It will not be present near the kinetochores as such, thereby maintaining the sister chromatids together. So again, at same diagram I have repeated as such, where it shows how the activity of the APCC would first be reduced, then it will increase, then again decrease and increase simultaneously. And most of our models from this, they have come from yeast as such, the fission in the yeast and budding yeast they have come. And later they found out that all these molecules are present in the mammalian as well as the human oocytes, thereby correlating that similar functions happen in the higher animals as such. So in addition to APCC, there are other cytostatic factors, the details of which we are not going to go into it with the time not limiting as such. You can see this is the APCC and complex, and so many molecules, they have a converging pathway on what activates or does not activate. One of the two things which I would say important is the CDC20 activation as such over here. And one more is the EMI2, because EMI2 is the one which is usually normally activated post-fertilization and results in the activation of the APCC complex as such. So what is the message that we are going to take over here? Usually a total failure, though it's so many steps are involved, it's a rarely it will be happening. Oocyte maturation defects as such can be a cause for infertility in what is otherwise earlier known as unexplained infertility. We have seen very few cases, but where it was all the oocytes probably at the GV stage or the M1 stage arrested and not progressing forward. And though these are so few cases, maybe they'll help us know how the physiology works, how the cell division works in future, and how we can overcome this factor. And maybe these oocyte maturation factors, what we poorly know of, probably what we call as oocyte factor as such, can be treated properly. And coming to the molecular parts as such, usually you have the checkpoints, which are much more tightly controlled in the mitosis, next during the spermatogenesis, and they are relatively relaxed in case of oocyte generation as such. Thereby, your fully grown oocytes are subject to less of these controls. Your probably mitotic spindle, the meiotic spindle formation is usually present as a good checkpoint, but still it is not a very good checkpoint like in mitosis. So what happens with this is, since the checkpoints are relaxed, they can happen a high frequency of the oocytes, which will be having the errors, and still they might be able to progress to a meiosis 2, and they can be seen as mature oocytes, which will be using. But post-fertilization, we see that the development is compromised for these oocytes. Thank you.
Thank you, Akash. Thank you for a wonderful and actually a very thoroughly researched, presentation of yours, right from the diplotin stage to the metaphase 2. What I personally think here is, from the embryologist point of view, all these checkpoints that you mentioned are absolutely important to understand, the oogenesis part of it, and why there are in particular cases, we get, as you mentioned, very few complete fertilization failures or other abnormalities as such. But from the embryologist point of view, I would like to know, and I would like to tell you that all these checkpoints that you mentioned, they occur in vivo. So neither the clinician nor the embryologist has any control sort of on it to overcome those checkpoints. And I'm sure in this webinar, maximum people are the embryologists, and they would like to know few points where occurrence of such abnormalities, how to overcome that once you get it, because they have to give answer to the clinicians as well. Of course, clinicians also must know about all these practical things. So when they ask the embryologist that why the fertilization is not there, or why particular oocytes you could not give the blastocyst to us, there are certain things where we observe, as an embryologist, we observe. So I would like to put two questions for you. This is slightly extension from the metaphase 2 and from the own pickup point of view, but it is certainly correlated to the previous stages that you mentioned for the checkpoints. So the first point that I would like to ask you is, how do you explain higher incidences of the polyploidies in subsequent and consistent cycles? Do you have any kind of checkpoint during the oogenesis as such? And second point, what I would like to practical from and important from the embryologist point of view is, can you correlate any cytoplasmic organelle indicators for the incompetent oocytes?
So as far as helping for the patient is concerned, like you have rightly said, these are most of the things which happen in vivo. But maybe, many a times what we call as unexplained infertility or unexplained, maybe the oocyte factor, when you change one of the gamete, which is the oocyte, and everything falls into place. So probably these are one of the potential areas where the oocyte is wrong, and maybe we'll have better answers, maybe not able to treat from an embryologist perspective, but maybe treatments may be there from the clinical perspective in future, not for now. And coming to the other two questions as such, what are the changes in terms of polyploidy checkpoints? So in terms of polyploidy as such, we know that polyploidy is not very well controlled. We have seen that extrusion of the polar body usually happens quite well in majority of the oocytes because of the meiosis resumption and the checkpoints which takes place because the spindle assembly complex should be there. But if you are not going to see a meiosis, the polar body in the previous as such, so that's one indication that it may result in polyploidy, the proper extrusion. Another one is in cases of giant oocytes where we see them, where probably there's a polar body which is present in the previous as such, but then these are polypoid nucleus over there, which again can be many causes of it. It could be an extra round of duplication over there. It could be endoreduplication which can happen, or maybe the centrosomes complexes which are formed as such could be multiple present over there before fertilization. And post-fertilization, it could be some problems from the sperm side as such, maybe multiple centrioles as such. Again, endoreduplication comes into place. So polyploidy as such is very tightly controlled, but yes, there can be certain times when it is not controlled. And we even know as such that polyploidy is present in the trophectoderm cells in a later stage when they are going to give rise to the placenta, when they form the syncytial trophoblast cells as such. And second question, coming to the second question, which was about cell organelles as such. During the development, if this pertains to the cytoplasmic part of the maturation, where we usually don't see the maturation happening majority of the times, but there could be certain indications that it has not happened properly, which could be like the mitochondrial distribution may not have happened properly. Maybe the organelles which are clumped together, they results in what we call as increased granularity present over there. And in these cases, then obviously when the cytoplasmic maturation has not happened properly, then in these cases, though the fertilization may be present, the progress of the cleavage stages would not be proper, and we see increased cases of, say, fragmentation, poor quality embryos, or blastocyst arrest as such. So I think this is one thing which has come to my mind.
That's true. What about the SCRs or
SCRs, yeah, SCRs could also be considered as a part of it, or you could even see as vacuolatory changes, where it is the cell which is trying to, discard the things which are probably toxic or not useful over here. You again have granules, you have pigmented particles, which could be a remnant of these things as such. So yes, cytoplasmic changes we do see where the cell is trying to overcome, but then nearly 50% of our oocyte would have one or the other abnormality in terms of the cytoplasm. So we can't say that having an abnormality is equal to a poor quality oocyte as such. But when the abnormality crosses a threshold, when we see gross abnormalities or majority of the oocytes are going to have the similar abnormality in large proportion, then yes, then that's a point of concern. And probably this is at what we do is at this time we inform the clinician and probably take a guarded call on what the prognosis is going to be for such a patient.
So in this case, you think that next cycle, change of stimulation might help?
Yeah, change of stimulation can help two ways. And I think I got it. Like one thing is even your trigger timings and everything could be there. And usually they say post trigger and the timing is sufficient. The podocytes of the surrounding cumulus cells are the one which will be retracted from the oocyte as such, allowing for their proper expansion and proper growth and resumption of the meiosis as such. Change of stimulation, yes, we have seen few cases where definitely a change of stimulation, maybe change the protocol, change the duration, check for your LH levels, which is often missed, check for whether there's a premature rise of LH or progesterone over there. So these are the things which can be modified from the clinical end, and they do result in better oocyte quality as such in the next cycle. Maybe it is just this particular cycle, or it could be there's a genetic orientation to it in terms of your receptors may be modified, or there could be something as polymorphisms over there that is still open to debate.
Correct, absolutely. Thank you. Thank you very much, Akash. And over to Sanket.
Sir, excuse me. Sir, I have a question here, sir. Just to extend your question with respect to polyploidy, and just to add a few points to what Dr. Akash has said. So basically, with respect to humans, basically, like, see, the attachment of the spindle fibers with respect to kinetochore is very specific. Like basically, like they are amphoteric in nature. It means like, see, first there is a chromosome here, so like chromatids here, like daughter chromatids, sister and non-sister chromatids. The binding of the spindle fibers happens non-randomly. Like say the centrioles is once dispersed to the polarity like north pole and south pole, and when they spread the astral rays or the spindle fibers, they're going to attach to a very specific points, like one to each. They're not going to cross over like this. They will like I don't know what cues they get and how this happens in nature, but they're very specific in nature. And they're gonna, when, as I mentioned, one of his slide, Dr. Akash mentioned with respect to separases, like this cohesins are the things that are gonna bind the chromosomes. So when this cohesins are degraded, the tension is released. That's how the chromosomes get separated, like they pull to each other, like to north and south pole.
Absolutely. That is true, Sanket. So these are not very common incidences, but I was talking about those rare incidences where in few patients you consistently see a higher incidences of the polyploidy. And as Akash mentioned, the polyploidy can be both ways. It can be female genome oriented or the male genome oriented. Both reasons can be there. So is there anything genetically involved into this where the checkpoint of either of the checkpoints we can predict it, and because of this the polyploidy incidence is high? Because this is to give answer to the clinicians as well as to the patient, because patient will also ask us question that why my oocytes are getting continuously abnormal fertilizations. So this is from that point of view, not from the practical point of view as well as from the research point of view. That is why I asked.
Sir, if I can add, like there's a difference like polyploidy and aneuploidy. So polyploidy is where the entire thing would be duplicated. And in terms of aneuploidy, what Dr. Sanket has already told and I told is the separation of the sister chromatids as such. And usually, as I mentioned during the talk also, during the mitosis, this SSC is very active. It's very tightly controlled. What happens is once this kinetochore, usually you'll be having two sister chromatids. I know it's a bit difficult to explain here, but suppose you have the two sister chromatids and in the center you have your centrosome. What happens is the kinetochore proteins usually are present throughout during the initial phase.
Absolutely.
During the later phases, it is from the chromosomal arm, they get dissolved, but are present only in the centrosome, the kinetochore proteins. So that's how the anaphase will attach specifically only in the center. And thereby, if it is two, one to one polar and the other to the other pole, which is bipolar, like Dr. Sanket already told. And then there's the APC, which cuts like a knife, the cohesion which is present. And then there's a pull, the tension which is applied over here. And that is a phase, that is a cue for the cell that the tension is present in all the chromosomes as such. None of the microtubule is not having tension as such. So all the microtubules are attached and there is a proper attachment formation of the metaphase plate in the center. And then the anaphase can start. That's the anaphase trigger for it. And the cells will, once this is achieved, anaphase happens just like that. It's like 10 to 15 minutes, it finishes, and then your telophase and cytokinesis happens within minutes.
Right. That is why aneuploidy you can see even normally fertilized 2PN when you see. Yes. That is why I was not concentrating on that part of it. I was just concentrating from the where you can clearly see 3 or more pronuclei.
I mean, other than polyspermy, I'm not sure of any genetic points as such.
So just to add, yeah, Dr. Akash, just to add, see, when you talk about polyploidy, we see a lot of clinically molar pregnancies.
Yes,
see, molar pregnancies just runs in certain people. If there's a recurrence, there's always a recurrence. So that shows there is some genetic predisposition because that is also kind of a polyploidy, no?
Yeah, molar pregnancies are mostly 3PM.
Sets of,
they found that it is of mostly of the paternal origin is what they have found, if I'm not wrong. And could be polyspermy.
Could be either. It could be maternal or paternal origin. And, usually we do see that there is certain women who are always predisposed to get molar pregnancy. And it is also sometimes seen with ethnicity. The Asian women are more predisposed. So as Dr. Vijay Mangalu is saying, there must be something with genetic when it comes to polyploidy. Though we are not very clear.
Factors we don't know as of now, like what is the exact genetic factor, we don't know of yet. But yes, they should be, like if it's going to run in families and ethnically, there should be some factor. Yes.
Yeah. So, Vijay sir, moreover, like polyploidy with respect to the status which is concerned, we get to see more in plants, like say rice, Pisum sativa, or your beans plant and your maize and all. Like it is very rare to see in humans, especially vertebrates. The incidence is very less, so that the mechanism whichever is responsible for this determination is highly conserved.
Okay. So that means in plants, the self-repair mechanism is much efficient than in humans, you say?
Humans is more efficient than in plants. That's what I would say, because polyploidy is very common in plants.
Okay.
And one thing I would like to highlight is with polyploidy, you can't detect by your NGS platforms because it checks for copy number variation as such. So you'll be not, if you are going to do a PGT and with NGS platform, polyploidy is going to be missed as such.
That's the limitation of platform.
Limitations of various platforms.
So how we can diagnose these things? Now, if there is problem with that, these checkpoints or things, how can we diagnose that? What is that?
I think probably the older one, like FISH, is the one where you can get a multiple signals. If you are more concerned, maybe a patient in whom repetitively we are seeing polyploidy as such, probably FISH is one of the techniques which you can still apply over here. But how that would be done, I'm not exactly sure in the present scenario where we are doing day 5 biopsies as such. Because that would apply more for single cell, and that has to be a day 3 where we can clearly separate the cell number as such. So they mentioned very clearly that it checks only for copy number variation, what the PGT, what we are doing. It checks for copy number variation. It can't check for the polyploidies as such.
And I don't even think it checks for indels, insertion and deletions as such, with respect to CNVs and SNPs. Anyways, with the permission of Vijay sir, last question to Dr. Akash. Dr. Akash, it was an excellent talk. And whatever I spent for almost 6 months to 1 year during my master's studying this cell cycle regulation, it's a part of my curriculum. So yeah, very well explained. And the point is, in one line, if you want to, few lines, maybe one or not one line, 2 or 3 lines, if you want to sum up what is the major difference between checkpoints in mitosis and what is the fundamental difference in checkpoints in meiosis?
The major fundamental difference is the additional steps which I had highlighted in one of the slides. So those 3 points, no S-phase division as such, and the prolonged prophase and the recombination proteins as such. So that is the additional checkpoints which the cell has to undergo. And one more major difference would be in terms of where, how these checkpoints are relaxed in oogenesis. So thereby they allow these defects to creep in. And probably that's one of the main reasons why human reproduction and human oocytes as such are, it's not a very efficient process in terms of what you get a mature oocyte, still could be aneuploid or otherwise. And probably these are relaxed. What is the evolutionary basis for it? Probably I could not think of. But then the final point is these checkpoints are relaxed, thereby creeping in of the defects, and thereby your reproduction as such is not very efficient. And once your reproduction as such is not efficient, your IVF as such automatically becomes not efficient.
So you mean to say,
of each oocyte.
You mean to say this is modified cell cycle regulation on part to mitosis?
Pardon? Modified cell regulation and?
On part to mitosis.
And part of mitosis, yeah. It's like modified cell regulation when compared to the mitosis. And there was one, what I mentioned, 1268 genes, they found it different between replicating cells within the same testicular tissue itself based on whether it is somatic origin or germ cell origin. So that many different genes needs to be present for differentiating mitosis and meiosis. So, I mean, it was a big learning curve for me as well. I come from MBBS background and I would have never read anything about these things except for something called as APC complex. And that's it, maybe the cyclin and CDC and phosphatases, and these results in cancer. So thank you for this wonderful topic. And I think I learned quite a lot while preparing for it as well.
Let's move to another question. Yes, Ananta, please.
Yes, sir. Can we have the poll questions? The next 5 questions. Most of the questions will be related to the first session, very few from the second one. So for the audience, the first question will be, every month the oocyte arrested in meiosis will enter meiosis 2 after puberty. True or false? That means the oocyte during every menstrual cycle arrested in meiosis 1 will enter to meiosis 2 after puberty. So you should say whether the answer is true or false. Coming to the second question, LH surge during ovulation causes dash. Decrease in cyclic GMP, increase in cyclic AMP, decrease in cyclic AMP, both A and C, sorry. First option is decrease in cyclic GMP. Second option is increase in cyclic AMP. Third option is decrease in cyclic AMP. And the fourth option is both A and C. Coming to the next question, dash is responsible for DNA repair during meiosis. Dash is responsible for, sorry for the spelling mistake, for DNA repair during meiosis. Answer option 1 is cyclic AMP, option 2 is cyclic GMP, option 3 is BRCA1 gene, option 4 is BRCA2 gene. Dash is responsible for DNA repair during meiosis. Options are cyclic AMP, cyclic GMP, BRCA1, BRCA2. Coming to the next question. This usually it will be covered in the next webinar. If it is very simple question, so if you know, you can answer. At the time of fertilization, oocyte development is resumed from. So resumed from. So we have covered till this stage today. So the options are telophase 1, metaphase 2, anaphase 2, prophase 1. At the time of fertilization, oocyte development is resumed from. Resumed from means what stage it is till fertilization. Options are telophase 1, metaphase 2, anaphase 2, prophase 1. And the last question is cyclin B, cyclin dependent kinases form dash. Cyclin B, cyclin dependent kinases form dash. Options are meiosis arrest, mitosis arrest, maturation promoting factor, prophase arrest. Cyclin B and cyclin dependent kinases form dash. Options are meiosis arrest, mitosis arrest, maturation promoting factor, prophase arrest. Can we see the answers? Can we have the poll response?
Yes, ma'am.
Yeah. So the first question is, every month the oocyte is arrested in meiosis 1, will enter the meiosis 2 after puberty. So the right answer is true, because during every menstrual cycle, it is starting from the meiosis 1 to, and it will enter to meiosis 2. It is the right answer is true. 75% of the audience have answered it correctly. And coming to the second question, LH surge during ovulation causes dash. It causes both A and C, that is decrease in cyclic GMP and decrease in cyclic AMP. Coming to third question, dash is responsible for DNA repair during meiosis. Answer is BRCA1. It is the right answer. 65% of the 67% have answered it rightly. Coming to the fourth question, at the time of fertilization, oocyte development is resumed from metaphase 2. So the right answer is metaphase 2. 50% have answered it correctly. Last question is cyclin B and cyclin dependent kinases form the maturation promoting factor. So the correct answer is maturation promoting factor. 67% have answered it right. Thank you. Thank you so much. We can move on to the panel discussion. Sangeet sir, you can take over.
Requesting the coordinators to introduce the
Yeah.
moderators and panelists. Yeah.
Ananta, you have to.
Yes, sir. Next, sir, next session will be on panel discussion about the handling of oocyte. The moderators will be Dr. Ved Prakash sir, who is a scientific director of Sperm Life Advanced Andrology Lab, Delhi. Already sir was introduced. So coming to the next moderator of the session, Dr. Sankeet Dumal Satya, the senior consultant embryologist, Hedge Fertility, Hyderabad. He is also an ASHRAE certified clinical embryologist. He has completed his MSc in genetics, MSc in clinical embryology, and PhD in reproductive biology. He has won so many awards, and he is a member, editorial board member of a journal, JBRA, Assisted Reproduction, ACE Newsletter, and IHERA Newsletter. He has been a reviewer in an international journal and authored many publications in national and international journals. Over to you, sir. You can introduce the panelists.
Thank you so much, Dr. Ananta, for the kind introduction. With sir, I would like to request you to introduce the panel.
Sir, you are muted, sir.
Our first panelist is Professor Dr. Sanjay Shukla. He is Senior Consultant Clinical Embryologist, Adjunct Professor Clinical Embryology, Department of Reproductive Medicine and Surgery, Mahatma Gandhi University of Medical Science and Technology, Jaipur. And next is Dr. Charudat Joshi. I think he no need to introduce. So many times we have introduced already him, but he's a very experienced embryologist, more than 20 years experience, and he is doctorate in life sciences, medical director, Genes India ART Bank, Indore, and lab director, IVF unit, RD Gardi Medical College, Ujjain. And next is Dr. Durai P, working as a chief embryologist at Krishna Institute of Medical Sciences, Hyderabad. Since 2009, he has many international papers on his credit. He has more than 17 years of experience in clinical embryology. He also visits West Africa as consultant embryologist. He is serving as an executive committee member of ACE. He had received Best Embryologist Award from Mercedes-Benz Health Summit 2019. Next. Dr. Prag Nandi, Scientific Director and Clinical Embryologist, Cradle Fertility Center, Kolkata, and he is a former research fellow, University of Kolkata, currently Scientific Director at Clinical Embryologist, Cradle Fertility Center, Kolkata, founder and life member of Academy of Clinical Embryologists, India, creator of ACE official logo, and he's a member of so many society and is very active. And he has published one newsletter on ART guidelines. Next, Dr. Meeta Sharma, she is ASHRAE certified senior clinical embryologist, PhD in medical microbiology, more than 10 years of experience in clinical embryology. She is certified embryologist to perform profectoderm biopsy, faculty at various national conference and workshop, author and co-author of research paper and book chapter, and she is a member of ACE, IFS, ISAR, ASHRAE, and past executive committee member of IFS. And Dr. Amita Subramaniam, consultant senior embryologist, Cloud9 Fertility, T Nagar, and she worked at Srusti Fertility Center, Chennai, and Santati Center of Reproductive Medicine, Bangalore, and she is a member of ASHRAE, ACE, IFS, and COGI IFS certification of embryology. She has done in November 2020. And she has had special interest in advanced sperm selection and oocyte dysmorphology and ART outcomes. Next, Dr. Nancy Sharma, senior consultant embryologist in Department of Reproductive Medicine, Jindal IVF and Sant Memorial Nursing Home, and she is ASRM certified embryologist, and she has done embryo and blastocyst biopsy training from Embryology and PGD Academy, UCL, and done embryology certification course from IFS, and more than 9 years of experience, and she is a member of so many society, and regularly she is presenting papers in ASRM and ASHRAE. And Abhinav Dudeja, he is MSc Clinical Embryology and PhD Senior Embryologist at ART Fertility Clinic. He has been a part of IVF industry for more than 5 years, and he has worked in Fortis Hospital as a senior epidemiologist. Co-author a chapter Lab Protocol in Hepatitis C in the Handbook on ART in Viral Infection 2021. Best Independent Thesis, Developmental Potential of Oocyte in Master. Invited as a speaker in various national conferences. Area of interest is troubleshooting sperm-related research and cryobiology, and is a member of ACE, ISAR, and ASHRAE. Sanket, you can start now.
Yes, sir. Let me share my presentation. Is it visible?
Yes.
Yeah. Well, as Vets has already introduced the panelists, which we gonna discuss on the topic of oocytes handled with care. So we all heard the past two lectures, which spoke a lot about how the human largest cell, that oocyte, is being produced, and what are the internal factors, like say with respect to checkpoints and other genes, the thick ocean of genes and proteins that are involved in making up this huge cell. So once this is harvested, as a being an embryologist, like we should know how this to be handled at utmost care in the lab. So having that background, and I would like to proceed now. And yeah, yeah. So as I said, with all this hard work of various hormonal influence and various cascade of events, like starting from queues and checkpoints and duplications and divisions and all, does this oocytes are really, you know, destined to face insults in vitro? The first question. Or are these oocytes are meant to be subjected to insults during IVF procedures? And finally, like, how to address these insults, like that oocyte, after like so much of hard work, like say with respect to males, like we are in millions, like sperms are millions, and but oocytes are like very like countable, like yeah. Apart from jokes, yeah. So like they're very precious, right? Even single oocyte is very precious, that might end up in a healthy pregnancy and healthy live birth. So what are the insults this oocyte is going to face in vitro conditions, and how to address these insults with respect to in vitro conditions, and like how to handle them for the proper or the better outcomes. Start before jumping into the panel. I would like to raise few questions, like you may call it as riddles in research, like how these oocytes of humans and mammals adapted to this internal fertilization process. Like we all know that there are two different type of fertilization process, like external as well as internal fertilization process. But how are these humans or mammals adapted to this particular mode of fertilization called as internal fertilization? And also, like, there should be some primordial reason, right, for this very strict reproductive dichotomy, like egg laying versus live birth, that is oviparous as well as viviparous. And if you look at it, yeah, egg laying or live birth, how does this evolution chooses? You can see fish, which is very much adapted to the external fertilization, and you can also see humans, where the internal fertilization is a method of birth. So when and why did this live birth evolve? Having said this, of course, research is yet to answer these questions. With this background, I would like to jump to the first question. Are these oocytes are sensitive to this light? Of course, yeah. Humans are tend to work in the visible spectrum. Like they need a light to see something. Otherwise, they're like blind. So we need light to see something. So when we are working in vitro conditions, like we do harvest these oocytes and we handle them under the light. But what about oocytes? Are they sensitive to this oocyte? Oocytes are sensitive to the light. Anyone can from panel can take up this question. And also, I would like to request Vijay sir as well as Dr. Akash, if you're fine with it, please join the panel. Since we're lacking two of our members because of whatever reason, so we would like to request Vijay sir as well as Dr. Akash to join the panel. Yeah, please.
Thank you, Dr. Sanket. Would be here.
Thank you. Durai sir, Meeta ma'am, how does this light affects the oocytes, especially in vitro? Because they're not destined to expose to the light, right? Whatever happens in vivo conditions, like they don't see the light at all. But here it is a different aspect where they are exposed to light. So how about this? How does this light affect the oocytes?
Yes, I think, Sanket, that minimum shorter wavelengths of the light, they do have the effect on the oocytes. So what we say that minimum exposure, specifically, I think microscope blue light should be filtered because it is having the effect. It may lead to the generation of H2O2, and that is obviously harmful to the oocyte. So the prolonged exposure under the blue light should be avoided.
True, agreed, ma'am. Others, please.
I think it's
As Dr. Sorry.
Yeah, please go ahead. No problem.
Yeah. As Dr. Meeta said that the exposure of the blue light should be avoided. I feel that if the duration of the light exposure to the oocytes is in a longer duration, that could also cause some sort of oxidative stress to the oocyte that will impair the embryonic development. But I have a follow-up question, if I may ask. In time lapse, there's a photo being clicked in every 10 to 15 minutes. What would be the impact of that light when it comes to the embryonic development?
Yeah, I wanted to keep this question in the panel, but then again, due to the time constraint, it couldn't keep the question. Anyways, yeah, as you mentioned, the light, it is going to take the images every 10 minutes, right? Like 22 hours, it is taking multiple images at intervals of 10 minutes. That means the fundamental point here is the light intensity which is used in time-lapse imaging is minimal, and it is very much adapted to the quality, whatever the ensuring the safety of the oocytes or the developing pre-implantation embryos.
Can I add on this?
Please.
Yeah. I think one thing was why blue light, because that's the higher energy wavelength, and more importantly, it's the absorption spectrum of your DNA and RNA peaks around 260 nanometers, usually in that range. So that is the UV light spectrum. So the more you are going towards the blue light, then the more the chances of absorption and DNA damage as such. So that's why I avoid blue light. And the second thing was for the time lapse, the question which Abhinav put. I think there are certain papers which have come up and they have calculated that the entire exposure, what the oocyte and the developing embryo would be exposed to the time lapse during the time lapse would be hardly minimal, like hardly anything. And it is not continuously exposed. And the recent papers, what they have come, yes, light seems non-physiological as such, as far as exposure to oocytes is concerned, because if it is in vivo, they would never be exposed to it. But what they have been finding recently is light may not affect that much. I mean, your normal low intensity light in the yellow part of the spectrum may not affect the oocyte or the embryo development per se, but then the lesser the better, not at the cost of making mistakes in the lab, definitely.
Yes, Nancy.
So I was also about to say that the toxic impact, and it depends upon the wavelength intensity and the duration of exposure. So when we calculate that whatever we are doing outside when we are seeing the embryos under the microscope, there are 95% of exposure is due to that microscopic light only. But when it is inside embryoscope, the exposure and the wavelength is lesser. So there are many papers which quote that and which associate that when it is inside the embryoscope, the exposure is lesser as compared to when it is outside. So what we can do is we can reduce the exposure time every time, or we can just be more quick and more precise with our grading system whenever we are doing it to avoid the light.
So what is the wavelength you required for that we can use?
The recommended wavelength.
Recommended wavelength.
The recommended wavelength, it's see, when anything which is upper than 300 nanometer, it's a UV absorbed. When the UV is absorbed by the plastic in that, and when it is more than 300 to 400 nanometer, it is going to affect the oocyte or the embryo, and it is going to affect it in an apoptotic way. It will affect it in that particular system that it started to it starts with the cycling of the necrosis, and so the wavelength should be lesser.
If I ask
anything above 500 plus should be good, where it results more in lesser of blue light and more of yellow line. And I think green filters are the one what we commonly employ as such in our microscopes.
For the light source.
Or the ICSI from the light source and till the oocytes or the embryos get exposed.
So here I want to add something. So the source of light. So nowadays we are started using LED source and fluorescent. Previously we are using fluorescent light. Now we are using LED. It will affect anything?
Sir,
it will be there in the source of light?
Yes, sir. The point is, sir, as far as me is concerned, like I would say the LED, again, the intensity matters, like how you're designing your LED. Say, for example, you're keeping the intensity with respect to lux, whatever angstroms, that is more important because the whatever is, as Dr. Akash mentioned, like visible spectrum, that is more convenient because it is going to absorb a lot of UV rays. Of course, it comes with a filter again, like you need to use a lot of filters like coating and other filters, which will absorb these harmful radiations and will emit only the safe light. Yeah.
Can I jump in?
Yes, Rahul.
Am I allowed?
Yes, you are allowed. Please go ahead.
Yeah. Just to add a few lines here, as the question was there that how does light affects? Basically, the light induces property of photooxidation, and this photooxidation may interfere in the lipid bilayer membranes, and it can eventually end up in photooxidation of DNA as well. So this is how the light can affect oocytes or the developing embryo. And the second question, which was related to time lapse, basically there are there are many journals which are quoting that a light intensity less than 590 nanometers of wavelength, these less than 590 is onto the safer side. So how the time lapse incubators are made, that they reduces the wavelength, and at any time the light is not on all the embryos at a time. It is calculating, or it is analyzing or taking photograph of only one embryo at a time. So all the embryos are not exposed at a time, only one embryo for 20 minutes. It is there for the photography. So this was my point to cover up.
Excellently covered by all the panelists. Like, yeah, first and foremost thing is, yes, you need to use the light, which is very much suitable for the gametes, especially in the in vitro conditions. And yeah, with respect to time lapse, yeah, there is the machine is designed in such a way that it will emit only the safe light, which is safe for the gametes or developing embryos. And then the frequency is very much calculated, and it is not like for all. It is like embryo wise. So it is very much optimized. Having said that, yeah, if you look at it, a lot of experiments have been done clinically and at a lab level. And what they found out is, if you are using constant bright light exposure to this oocytes, they gonna increase the incidence of spindle or chromosome, say mitochondria, also no aberrations, and also also the mitochondria contents will be disturbed because of a lot of ROS is generated because of Dr. Mita said and other panelists said, a lot of ROS is generated because of exposure to the light, which has to be addressed. For that, this mitochondria has to function a lot. So that is another thing, and where also there is a epigenetic level, there is also differences with respect to acetylation, methylations, and demethylation levels, which is very specific to each kind of gamete. And also, see, basically the constant light exposure is not advised because it might end up in lot of oocyte meiotic defects and also quality deterioration, especially the lot of studies has been done in mice. And also, if you look at it at the lab level, especially in the human oocytes, say a very classical flowchart which has been put up by Mortimer, where see, for example, hospital management has installed a new fluorescent lighting throughout the hospital, and say including IVF lab, and also suddenly the pregnancy rates have dropped. So what could be the reason? So how could you describe this issue? So basically, it comes under the light category, where what is the strength of light emitted and how can the gametes and embryos are protected with this light. Secondly, how to resolve this is like install a dimmer switch where you can control the lighting level. Like say, whenever you want, whenever you're getting the gametes or embryos outside the incubator, you can dim the light, and so that is where the functionality of this dimmer comes in place. And also, like have a consider having a fewer lights switched on when the processes are going on in the lab. And also, finally, you can have a protective layers around the light fittings. For example, microscope fit. It could be microscope fittings, or it could be ceiling lights, or it could be inverted microscope fittings, or wherever the light source is coming up. It is always suggested to protect these light source with the protective filters that ensures the safety of the gamete. So basically, light levels are optimal for ART. This has to be considered. And yeah, a lot of studies, another supporting data which I want to show up is like, see, a lot of studies have been done in the rabbit oocytes, hamster oocytes, mouse oocytes, and what they found out is like whenever the amount of time is extended or like increased to the exposure to the light, there are detrimental effects that could be like cannot be corrected. So it's always better to minimize the light conditions in the lab as far as when the gametes are handled outside the incubator. Now, I would like to ask, request Ved sir to take up the second question. Ved sir?
Sanket, my sound is not coming properly. I think you have to ask
No,
me.
so we can hear you.
You can, yeah, then it's okay. Akash was telling me, no problem. So how, as you know that we are doing that denudation at the time of ICSI and before that thing. So how toxic is high urinary enzyme exposure when we are doing denudation before ICSI? So what is the exposure time and how much, how much oocyte you can take one time and what type size of drop you will use to denudation? That is the questions, I think. Next, there is any question other?
Yes, sir.
Yeah,
Follow question about the concentration.
concentration, yeah. And safety and efficacy.
So can I talk?
Yeah, please.
So the ideal exposure time would be around 30 to 40 seconds. More important about the concentration is most of these commercial hyaluronidase solutions comes in a very concentrated form. So depending upon the brand that you use, it's important whether the brand tells us to dilute the hyaluronidase or to use it straight away. So care has to be taken. If that is not done, if you use very high concentration of hyaluronidase, you might end up having parthenogenetic activation of the oocyte. Whatever brand you use, first check what the concentration is. Most of them comes in a 10 times concentration, which means you have to do a 1:10 dilution. And so safety and efficacy, the size of denupets that you use, too small size of denupets, you would end up rupturing the zona and you will have zona free oocyte and a ruptured zona. So care must be taken. So when you're training people, don't allow them to take all the oocytes at one time. Just give them a few oocytes and then denude. So and with good pipetting skills. And
and it must be very quick and done efficiently. Some people do it under oil, some don't do it under oil. So that depends upon what protocol you use. So that's it.
Can I add here something?
Yes, yes, please.
See, as far as my experience, I can tell there should be the minimum exposure time. Now it may be 20 seconds, it may be 30 seconds. It should not go more or it should not be very less. What I feel is by denuding, uh, once you start seeing the polar body, that is why you just denude it, right? So once you start seeing polar body, then there is no need unnecessarily to keep on denuding till the entire cumulus is wiped out. It can be left if some cumulus is left behind. There are papers which shows that it helps actually in fertilization. So that is one important aspect. You should not keep on trying to remove the entire cumulus. Once you see polar body, that is fine. Second thing is the denuding pipette, as you rightly said. I have personally seen people using 135, 140, 150 micron denuding pipettes. But what I observe is 135, 140, sometimes it's really very small one. Means there is degeneration of oocytes during denuding. So for me, what I observed is 150 is an ideal size, which can have some amount of cumulus also left behind, and you can very well do it. And the exposure, yes, exposure ultimately, and the speed at which you are doing that also matters. It's not only hyaluronidase enzyme concentration, it is how many times you take in and out and how fast you take it and out. That also causes the problem with the oocyte, maybe the spindle or the internal cytoskeleton of the oocytes. So as far as safety and efficacy is concerned, it should be as early as possible. Second take-home point may be the number of oocytes. As you are expert, you do denudation of those many number of oocytes which you can handle easily.
We have a next question with respect to number.
Yes. So these are few points which I could I would like to say here in this hyaluronidase enzyme.
Yes, Dr. Durai. Yeah, Dr.
Add
Durai.
something to it?
You want to add something?
So wait a second, can I add something?
Yeah, please. Go ahead.
As far as exposure time, like rightly said before by Charu sir and Dr. Mita, it should be as low as possible. 30 to 45 seconds, they usually keep as a upper limit. Does not mean that we need to denude compulsorily for 30 or 45 seconds in hyaluronidase. It has to be as less as possible and to an extent where each individual is comfortable, because post-denudation, the number of cumulus cells left over should not be interfering with your ICSI process as such again. Yes, few cells left over definitely would not interfere as such, but then again, each one to his own, they decide what capabilities they have. Next, for the concentration, the standard what is usually they say to use is 80 IU per ml as such. That is the concentration at which it is supplied for majority, or maybe sometimes 10 times concentrated where you need to dilute. But then what I have found personally is 80 IU per ml. You can go much lesser than that and with an equally effective denudation as well. Maybe 40 or maybe even 21:4 dilution. Even in those cases, we do find effective dilution. And if I'm not wrong, it was Arne Sunday who has said that he uses 8 IU per ml on a regular basis. 8 IU per ml for denudation. This was almost 5 years back when he had a talk, and he said, and I asked him, the next question was, how much time you denude for it? Like 30 seconds. That's it. Why increase the time? It reduces the concentration. But again, like I said, you need to be well versed with the techniques. Otherwise, you give too much of a change of pressure. It's not just the timing and the concentration, but the pressure with which you are going to release on the denuding pipette, the in and out, the motions which is going to be created, the shearing forces which are going to be there, those matter. And probably it is when it's in excess, that's when you are going to have a zone of free oocyte as such. Safety and efficacy, like already covered majority part. I think one concern is regarding if you expose over, it's at the end of the day, it's an enzyme. You don't know how it would act. It could have multiple points of action downstream as such. One of the things is parthenogenetic activation of oocytes as such when they are exposed too long to a hyaluronidase.
One more point I would like to add, if it's for ICSI, it's okay to leave a few cumulus cells, but when you're doing oocyte freezing, we need to strip the oocyte completely so that there is enough exposure of the equilibration solution, vitrification solution. So that point has to be kept in mind when you're doing oocyte freezing.
Yeah, others, Dr. Meeta, Dr. Durai.
I think majority of the points covered. So here,
Yes, I think, yeah, all the points had been covered by Charu sir and Akash, everyone. Yes, less exposure of time, and nowadays, as Dr. Akash has said, that even there was a paper published of reduced concentration of the hyaluronidase, that is 8-IU by Anne Sunday, that has been published also. So they say that if the concentration of the hyaluronidase is reduced, it doesn't affect the embryo development later on. So reduced time exposure and reduced concentration both will help.
Even if I want to add something, we used to do direct denudation initially 2 years back. We used to take 80 IUs per ml and we don't used to dilute it. But then we started diluting it and we increase the diluted proportion. Now it's almost 10 IUs per ml, which we are 10 IUs per ml, which we are using, and it is giving fantastic results from then. And the results are okay, and everything is working fine. But the thing is that initially the exposure time used to be like 30 to 40 seconds or 50 seconds. Now it is reduced to 20 or 30 seconds. So exposure time is also very important for that. And the denudation pipette which we are using, we always start with the 200 ones, and then we reduce it if we want it. Otherwise, as said by sir, that for ICSI, if you are able to see the polar bodies, it is okay to go with it.
Actually, sometime what happened if you left some cumulus with the oocyte, then during the injection time, sometime that oocyte moves suddenly. So you have to take care that how much cumulus is okay.
Is okay, right?
Yeah. So that point should be taken care.
So
Yes,
our
okay.
beneficial side effect of this would be the cost as well.
Yeah. Right.
Yeah. So yeah, very well summed up by all the panelists. Like it's wonderful. Say it is very much standardized. Like we normally use 80 IU per ml as a standard. It's all about enzyme kinetics, like the amount of enzyme concentration and the time of exposure, right? That is what matters. And like, yeah, you should be as soon as possible, as quick as possible. And after listening to Nancy's point, I want to add one more point here. See, like, of course, certain labs do follow that, like they cut the cumulus and they keep the culture. So it's all about that. Like if you're cutting it, it is always better to dilute the enzyme concentration before at the time of denudation. If you're not cutting it as such and you're using it like raw, whatever has been harvested, it's always better to go with the raw concentration, like what company has recommended. And well, having said that, going up to the next question, well, it is all about temperature. Yeah, it's a very important thing because it has a lot of effects on with respect to the oocyte and the outcomes. Uh, First and foremost thing will be the temperature has any effect on the oocytes. And the follow-up question would be, what are the ambient temperature for the oocyte handling in vitro? And what are these damages that are caused by any fluctuations in the temperature, especially in vitro conditions? Any of the panelists can take up the question.
Yes, obviously temperature do affect the oocytes, and basically the ambient temperature is 37 degrees centigrade. Below the 37 degrees centigrade or higher than 37 degrees centigrade, it is going to affect the first of all the meiotic spindle, that is damaged, and then the cryosskeleton, then maybe other damage. Basically, meiotic spindle is damaged by the alteration of the temperature, and then it may result poor embryonic development later on.
So 37 is crucial for spindle morphology, but 37 within the media is crucial. So if
Yes,
it's
within the media.
is
Yeah, it is
crucial,
within
right?
the media.
Yes.
In the dish in which you have kept the media, kept the oocytes, in this, that temperature is important. That 37 should be maintained.
And if it is more than the more or lesser, or if there is any sort of heat stress, it diminishes the oocyte developmental potential in many ways. Like there is a series of cellular and molecular damages which can happen, including the spindle disintegrities or spindle damages. So, and the exposure is also important. If there are multiple studies, I can remember there was a study which I used to like very much initially, because for 2 minutes, when the exposure is for 2 minutes, it can come back. But for 10 minutes, it cannot come back. Then with 30 minutes, it cannot come back. So this is that study I used to like very much at that time when I was learning.
Exposure 2 minutes followed by incubation for hour.
Yeah, half an hour. Yeah.
To come back to the normal position. When compared to like exposure for 10 minutes and incubation for 1 hour, you can't get the same.
Yeah. That is what I'm saying. That I used to like that study. I think that for 2 minutes, it is giving us all back. But if we are keeping it exposed for a longer time, it is not. We are not going to get what we are looking for.
It's like you're giving time to bounce back, and you're not insulting it too much. You're not insulting it too much. And one, I think what I found, no, usually ambient temperature, like rightly said, it should be as close to 37 as possible. Little lower side is okay. I mean, little lower is still okay. Like they say the tolerance for oocytes and even the sperm for lower temperature is much better than what it is for the higher temperature. Like you should never overheat it, because even 0.3, 0.4 degrees centigrade overheat is much more dangerous than something similar on the lower side as such, because it's the spindles which is most sensitive. Whatever most of the papers that we read, I think they tell about, one is the spindles depolymerization. I think the commonly, like what you just now said, Nancy, that the 33 degrees centigrade is at which the permanent damage happens, if I'm not wrong. That is what the paper said. And one of the reasons why it is much more important in oocytes is they have this mechanism of heat shock protein generations. It still can undergo transcription. Though we say that it is not active, HSPs can still be generated, while for sperm, it is not that much active, HSP generation. So probably that's the reason why oocytes are more sensitive to the temperature fluctuations.
Here I can add something. You are all talking about the temperature shock and everything, no? So I'm agree with all the points. But I think Akash may remember in Hyderabad, one of the centers, as one of the, I'm talking about the real case what we faced. One of the embryologists from Mumbai, they did the case, and I'm talking about 6-7 years back. So around 10 to 12 cases are there in the incubator. It's a Hiracell box incubator. Night around 11, 12 o'clock, the whole power was shut down. The watchman doesn't know about anything. Okay.
And there is no backup.
Morning, no backup. So there is no one is in the campus. So morning 7 o'clock, that somehow that watchman realized is there is no power. Then he seen through some window, there is everything off in the lab. He immediately called the clinician. The clinician called the Mumbai embryologist. He called me. We rushed around 8:30. We were in the center open. The incubator is off. Because in the we can see the time and everything, right? We can cross check that in Hiracell 150 IET. Yes. So we checked it. It's gone around 7-8 hours. There is truly power cut. It's on day 2 embryo. So day 1 done, day 2 it's running. So around 48-50 hours. We taken all the embryos are cleaved. But unfortunately, out of 11, that fellow got around 7 to 8 positive in that case. Did you imagine? But still, I don't know, it's maybe a miracle. But the same case I faced in Africa. The Africa, you know, all that's a worry. The power is very worst in that Africa. Still, it is not developed as an Indian. I have around 200 oocytes in my hand. There is total power shutdown. So I can't get any power. Only I can run the incubator. Microscope, I'm not able to get anything. But so for that case, I did all the ICSI without any power. You don't know that lighting, this is RI machine. So if it's nursing, I can't do anything. Because of RI, I saved light source with star something I managed. I could, without heating stage, I did around 200. Like you said, no, 2 minutes, 3 minutes. That's the maximum I can spend per case. Not I switch on the timer myself. I keep it. If it is 3 minutes, I keep it. That case, that much. If I have 10 usage as well, if it is 7, I keep it inside and not touch more than that. That is the one. So on that case, it's that means the gold case. So after that, the bond was very increased with the center because that is the maximum got positive in that batch. I don't know. It is working in the reverse way. Like Akash said, the transmission works in the my favor. Something happened, but that was the better. But I felt after the few incidents, I felt myself, the oocytes are more susceptible to the temperature fluctuation. It's not much affected until you are handling very fast. They are able to survive like in crash conditions for some time. But if you are dramatically manipulating in outside the inverter to keep not keeping 5 minutes, 10 minutes, it may affect. But unfortunately, it won't affect that much parts when your temperature. Sometimes they are telling the 2 degree not working in this XT stage is not right. It is not like that. It will work on, but still you have to be maintain the 2 minutes, 3 minutes gap. Nancy clearly said the paper. Really, that paper still I love it.
That paper was when I started. So that paper was fascinating. If 2 minutes it can bounce back, but if it is more than that's why I mentioned that paper.
It is 1995 paper.
That see, it was old paper.
Yeah, the whole paper, I remember the paper, I did the case 2 minutes.
I'm talking about somewhere around like 2014 or 2015 when I joined.
So if I can add to what like Durai sir has just now said.
At that time, everything was fascinating about this embryology. Like everything was like, wow, how it is doing, how we are doing this. Everything was like that.
Yeah, Dr. Akash, you wanted to add something?
So, I just would agree with what Durai sir has said to a good extent, because I think there's papers exact to something similar scenarios what he has said. And if it is by Thomas Rusty Poole, I think, where we have attended webinars of his, and he has multiple papers, and he has presented, and he said that try to maintain as close to 37 wherever possible. But in case if it is going down, like if you are in a SOS situation, then maybe the oocytes do tolerate better. It's not that compulsory that all of them are going to go bad and you're not going to have any result. But then yes, our chance of compromise is much more higher. And in this case, I think what matters a lot where the light got switched off, I think probably they had a box type incubator, because that helps a lot, because your fall is not very direct. Like the fluctuation degree is not very rapid. If you have a big box type incubator in this, it's not very rapid compared to a benchtop, where more or less the changes will be much more rapid, and you more or less lose the oocytes or the embryos which are present over there.
Yeah, well summarized by all the panelists. Thing is, yeah, I just would like to add one of my experience, what Doris has said. Sir, I also had experienced the same situation where this motherboard of this RI system got spoiled, heating stage. So when I requested the company, they said it needs almost like 1.5 months to get it. So all this 1.5 month I was doing cases without heating stage, like as you said, 2-3 oocyte inject करो, वापस रख दो अंदर, फिर 2-3 oocyte लाओ, inject करो, वापस रख दो, because we know the chemistry of like what is the drop in the temperature and how much osmolality changes will be there if you expose more than 2 to 3 minutes. So first thing is, of course, sperms are very much, thanks to sperms, they're very flexible. They used to adapt. First I used to catch the sperms and get the oocytes, inject अंदर रख दो. That is what I did, and of course, I remember I did around 15 cases by then. So almost
All
like.
men,
Sorry, sir?
all men, sir.
All men. Sir. So out of them, at least I got 50% result. I'm still happy for it. And yeah, as you rightly said by all the panelists, temperature is the most important thing, because any disturbance in the temperature is going to affect the internal quality of the gamete, especially the oocyte, where, as Dr. Mita said, there will be a disturbance with respect to the spindle assembly. And of course, Dr. Akash, Dr. Dore, Dr. Nancy Allah mentioned a lot of points where it has a detrimental effects, any fluctuation in the. So it is always better to maintain 37 degrees Celsius or the ambient temperature whenever you're handling the oocytes, whether it's with respect to the handling outside the incubator or especially with respect to the micro manipulation part. Having said that, yeah, suppose say again, this is a very standard picture, like process map where you're gonna troubleshoot whether there's any difference in the temperature control. Of course, I've extracted this from again from Elder, yeah, and Brian's textbook on troubleshooting. So if suppose there is a poor XC embryo quality since the XC station was moved out to the different part of the lab, which is very close to the air vents. So what are the best area, like possible reasons that you could describe there will be a difference in the outcomes? So yes, of course, there will be a temperature drop with respect to the media, which in turn changes the internal pH of the gamete, especially the oocyte, and it has like a lot of effects to follow, a cascade of events that are gonna represent. So here you must revalidate the heated stage with respect to the change in the background air temperature. And second would be you should reset the heated stage to compensate the temperature loss. For example, so suppose you have placed the micromanipulator under the AC vent or AHU, whatever the vents that are provided, there will be loss of temperature, right? So suppose say if you set the temperature, like set temperature is 37.3, then it is like showing the display temperature of reading temperature of 37 degrees Celsius, then you should go with it. So like you have to reset the stage to compensate the temperature loss. And third would be like consider relocating the XC station away from the air event if it is feasible. And fourth would be like you should always put a like if you try to you should try to put a protective shield around the XC station. So this is how you should take care of XC with respect to temperature. And yeah, there are again, this is a same picture from the same textbook where they've shown like what is the lower temperature, what is the ideal temperature, and what is the high temperature. So there are three categories which they categorized, and with respect to every category, like with respect to blastocyst development rate from 2 pm and also the implantation rate as well as LBR per retrieval, the best possible outcomes are from the ideal temperature, that is 36.4 to 36.9 degrees Celsius. Anything above it is like it's too hot, anything below it is too cold. So how much hot it is bearable, or how much hot is too cold, how much cold is too cold. So that is how I would like to sum up this question. And yeah, wait, sir, can you please take up the next question?
So there are so many stresses are there. We are actually, I think Sanket, you have to take these questions. I forget that thing. Flow is down.
So as we discussed something about the denudation part, like with respect to the enzyme, the hyaluronidase. But here we also use mechanical denudation, right? Followed by enzymatic denudation, we'll be doing a mechanical denudation, where we gonna use denuding pipettes. And the question is about this: what are the recommended guidelines and considerations for denudation of cumulus oocytes? And followed up question is like, what is the optimal number of cumulus oocyte complexes to be denuded at a time, and any other important factors or considerations that should be taken care to ensure the success of this particular procedure? Yeah, I think that I think Abhinav is sitting quite for a long time. Abhinav,
yeah, so I feel that you should always make sure that you are not using the concentrated hyaluronic acid directly. It is always better to dilute the hyaluronic acid in terms of while pursuing the denudation. And I also feel that the number of oocytes is very subjective when it comes to embryologist. It can vary. However, if I talk about myself, I take a 100 microliter drop, and I tend to take anywhere around 10 to 12 oocytes in that drop. I start with the 200 and go down to 175. I would say that I do not prefer pursuing 140 stripper tip until absolutely necessary. 170 usually does the work for me, where I can see the polar body and I'm actually able to inject the oocytes. I use 140 only when absolute necessary, because I feel that squeezes the oocyte a lot. and that could lead to some sort of changes in the structure of the oocyte, and in turn degeneration can happen while pursuing ICSI.
Then I would add some point.
Yes,
Number
sir.
of oocytes you are taking at the time. So when you are taking some more, for ideally for me actually 4 to 5 is the maximum. Some of them we can take 10 at a time if you are taking more number of oocytes OCC to denude, it will be harmful to the oocytes because if you are taking more number you have to give them more pressure. As Abhinav rightly said, 170 is ideal fit for all. When as Nancy said, when you are using the Amita said that when you are oocyte freezing, you need to use at least 140 for the small wash to clear all because the equilibrium should be far better than when you are humoral cells there. It will interfere the equilibrium time. So to avoid that kind of such a condition, for oocyte freezing, I use 140 for just to make sure all are gone out clearly. In other for regular my cases, mostly in 80% of the cases, I use only 170. It is more. I'm very comfortable. And moreover, when I'm buying, I don't buy that very concentrated branded Hylace. As possible, I buy a ready-to-use Hylace because that derivation, that dilution part, I don't like. Somehow I not feel comfortable. Which are available ready-to-use Hylace. That is easy. For my concern, I feel comfortable. So directly, I no need to concentrate. I know to calculate. So this is the concentration. What is that? And the time number of that normally 3 concerns will be there. One is the number of OCC how you are taking and what the pore size you are using and the time of exposure. It's enough for 3 points is major concern for me to avoid the stress on the oocytes at the time of pregnancy. Moreover, once you denude it, you should give the enough time to get rid of washout from the JAPI. So all the cumulus, the Hylace attached to it, it wash out to the media. Because sometimes we are in a rush, we'll start the ICSI within 2 minutes, 3 minutes. It's not ideal. At least for me, 40 to 1 hour should wait after the denudation. Then it will be the ideal to do ICSI. So because if it is sometimes it will start immediately the ICSI, it may possible to interfere with the hyaluron still may attach with the zona. I think that's my done.
I would like to add upon Durai sir's comment. When you're talking about guidelines, we also have to talk about the time interval between pickup and denudation to ICSI, because that also matters. People follow different methods and all of them seem to work perfectly. But if you have a standard practice in your lab, troubleshooting on that part would be easier. So that if you practice one method, one timing for one case, then it will be more complex to decode where things go wrong. Ideally, we do denudation around 2.5 hours post pickup and half an hour later ICSI. And number of oocytes, we do around 10 to 12, maximum 15, not more than that at a time. As discussed earlier, pipette sizes. Only for oocyte freezing, for that last brief thing, we use 140. Otherwise, we stop at 175. And we don't mind having a bit of cumulus during ICSI. We don't completely strip the oocytes. That's it from my side.
I think for the guidelines part, yeah, what I would say,
Guidelines,
we all started with one.
Guidelines.
Sir?
Sir, we make one guidelines. How to denude? We make one protocol for India. We unitedly this can follow. Whoever is willing to attach us, then you can consider us because there's a huge gap in India. Yeah. Everyone we are doing best work. I'm not denying. Yeah. I am doing best work. Everyone doing the best work, but in their own way. When you are coming on the track, everyone travel on the train, so 1,000 people in the all bogies will be in the same track. Now what we are doing, all on track, but different bogies are traveling. We are not, as a India, we are not having any whole data. One
Sir,
art
even within chains of hospitals, they follow different protocol in different branches. So there is no consensus
Yes.
anywhere.
It is individual about embryologist. You and me are different, like that.
I think what we can agree for is at least we all started with one OCC at a time.
Yeah,
We all started with one OCC at a time. The beginning point was one OCC at a time. At least that is the common factor, I guess.
That much only senior can give.
Yeah.
That is the
Yeah,
biggest
one
thing.
OCC. Yeah, and other than that, I think for the denuding pipette, I think we are more comfortable with even using 150 and 140. I don't find much of a problem as such from my end. But what I would like to add is whether it is 200, 175, 170, 150, 140, whatever you use, the first time when you are going to aspirate that OCC or whether the oocyte with surrounding cumulus, it has to be individual. You should not try to take the first time you are going to use that pipette freshly. It has to be one OCC is what I follow for myself. Obviously, there could be modifications to it so that the pressure changes are not very drastic. You know, if you take multiple OCCs at the same time for the first time in the same size, when you reduce the pipette size, the first one is going to get just stuck when it is going to inside, the pressure increase and it goes in. And if you are going to take another one at the same time with the first OCC still inside, that is going to harm all of them together. So this is what I feel that I think 140, 150, I rarely see a zona free as such for my end. From my end, I don't have a problem as such even with 140 or 150. Yes, as far as cumulus is concerned, initially I started off where we wanted that neat oocyte, nothing else cumulus surrounding it. But now gradually we have shifted to where few cumulus cells are present. It does not make much of a difference in the ICSI. Probably with more of the IVF and more of the literature that it's non-physiological, we are more comfortable with leaving some cumulus cells around during denudation.
Anybody else would like to add anything for this? Well, very well sum up by all the panelists. Like it is like the plethora of experience speaking in volumes, right? Like everyone is practicing a different, no, standard protocol for with respect to denudation. Like how to denude, when to denude, what are the sizes of denuding pipettes they're going to use at different labs, and also the number of oocytes, like COCs. I mean, sorry, the COCs they're going to handle at time is also important thing. It's all about like see if you're the number of the time, the amount of time if you're exposed, the exposure of this COCs to the enzyme is more, probably you can use the larger pipettes. If the exposure to the enzyme is less, then probably you have to use the smaller pipettes, because you cannot strip out all the cumulus. That is what I feel with my own experience. It's all about the enzyme kinetics and the mechanical pressure, the shear pressures which you're going to put on the COCs to strip off the oocyte. Well, having said that, yes. Well, next coming to the more important topic. With sir, would you like to take question?
Then you can continue. I can in between. I will come.
Yes, sir. So yeah, welcoming. It's about cryopreservation. So does cryopreservation of oocytes has any impact on the oocyte quality? Because nowadays we tend to freeze a lot of oocytes. So it has become a, like I would say, not standard of care. It's like alternate a procedure where we tend to freeze oocyte for various reasons. And also like I would like to ask all the panelists, what are the changes that are triggered by oocyte cryopreservation, and also like as we all know that we are exposed these oocytes at the time of vitrification to a lot of volume of cryoprotectants, like say a very high volume of cryoprotectants. What are these cryoprotectants like gonna affect? How this cryoprotectants gonna affect on a gamete level?
So I feel since it's the largest cell of the body, and since it's a single cell, there's a lot of water content inside the cell. So all that water content needs to come out, and the cryoprotector needs to go in for us to have a better post-thaw survival. And I feel that if that thing doesn't happen, it can lead to either degeneration of the oocytes post-thaw, and it can also lead to changes in the meiotic spindle. I've also sometimes seen that when I thaw the oocytes, there are some sort of vacuolation of the cytoplasm, which is of course transient, but that is some sort of changes that happens post vitrification as well. it definitely is dependent on the media that you're using, the skills of the operator that is handling the case, and also, of course, on the carrier that you are using, the cryotopsy, whether it's closed or open. So it does affect the quality of the oocyte because the osmolarity, which is anywhere between 272 to 290 in a culture media, is approximately increasing by 20x, which is around 6,000 in the VS solution. So there is a lot of changes that the oocyte has to undergo in that 15 minutes, 10 to 15 minutes time frame. So you need to be extra cautious to avoid any sort of osmotic shock, the injury to the oocyte, and also ice crystal formation in the vitrification process.
Agreed. Very much agreed. Yes. Anybody else?
So we do quite a lot of oocyte freezing, both planned oocyte cryopreservation and for cumulative pooling of oocytes for poor responders, like patients with 1 or 2 eggs. We do multiple rounds of oocyte freezing and then do a cumulative exceed further on. It's an ongoing study also for us.
Instead of that, you can do freezing. What you are freezing the oocyte?
Huh?
You can make an embryo and you can freeze, no?
There is a cost factor involved. Instead of charging multiple rounds of ICSI, we charge a few rounds of oocyte pooling and do one cumulative ICSI. So there is a difference in younger women and older women, obviously. So with respect to oocyte freezing, so it's there is no margin of error. It either survives or degenerates because it's just a single cell. So most commonly seen changes is we see hardening of zona, and then sometimes the shape of the oocyte also can be deformed, but it kind of gets resolved with incubation, and with respect to doing ICSI with frozen oocytes, we need to give at least 2.5 hours of incubation time before going to ICSI because the spindles to get aligned again. And sometimes I have seen that the polar body, the cytoplasm nicely expands after 2.5 hours, 3 hours, but the polar body is still yet to, we just see a faint polar body. That's very commonly we see yet to expand, or that alone gets degenerated in a few cases, especially in the older woman group. So that is one thing. And chilling injury and all this, because it affects the lipid membrane, and all this chilling injury happens at this 15 degrees to -5 degrees, and care must be taken during the vitrification process. The number of oocytes that you load in per device, depending on your capacity, how much you can load. You can't load everything in one crylog. So that also has to be taken into consideration.
Amita, I have a question for you. As you said, you have a lot of experience with oocyte freezing. Could you express, like, in terms of percentage, like what is the survival rate and what is the
We
degeneration?
have a pretty good survival rate, almost 98% survival rate, because I have my current statistics here. Almost 98% survival rate. Good fertilization rates and almost 45% blast relation rate, even in the older woman group.
Okay.
So
What
that
protocol are you using? Like, I mean, with respect protocol in the sense that with respect to media, what media are you using there?
single step.
No, I mean the freezing.
Vitrification, sage, origio media.
Okay, cool. Yeah, Doris sir, Mita ma'am, Nancy, anything you want to add?
As what oocyte freezing, no, nowadays we are mostly doing social freezing. So initially we are doing the oocyte cryopreservation. That for onco cryopreservation we are doing many. Instead of socio, we try to avoid, but lot of single women are now coming. Last year we are submitted data for in Hyderabad. We done around 10 single women oocyte freezing. Sorry. Here on the recently we educated the patient in such a way. If you are they were somewhat we promoted in the oocyte freezing. First of all, it is I'm not 100% comfort with it. Then for coming to the procedure, the selection of the dish, which dish you are freezing there. Always some of the people will try to freeze in the forward dish. It is not right. Always your dish should be concave. That is which is provided with the media, because when you are putting the dish, you have to add in the wall. That is go slowly. That is the equilibration will be in the very uniform. That is the best step you get your better survival rate. Initially, I struggled with survival rate, but now I'm getting decent survival rate. And after the main challenge in the oocyte collector, after thawing, not only survival rate, blastocyst formation and LV. So whatever your birth rate is important. But I'm getting decent, but still it is, I'm seeing my improves from the beginning. It's not up to the mark. Now it's recently reached the good blastocyst. If I'm doing 12 oocytes, at least 4 good blastocysts I'm getting. That much I can do it. And moreover, the Crayola you are loading, I load maximum 3, not more than 3 in single Crayola. I have 12, I use at most 4. So not in everything. So some other people will do 6 and 5, but I'm not comfortable. Maximum 3, I feel ideal. And when you have thaw also, you have to thaw 3. It will a lot of time it needs, but it will be the best result for you. Spend the time, I think you will get a good result with the oocyte freezing. The cryo product, and I think it's matching it. All the companies now standardized. Initially, last 5-6 years back, that oocyte freezing medias are not standardized. Everyone is a trial and error based. Now, almost all companies are doing the good that standardized media. So, I think that any cryo product will work for you. It won't make any difference as of now.
With respect to social freezing, most of the women who come for social freezing are already past 35 years, which means the oocyte quality is going to be poor. So how much eggs are you going to collect? There is no proper limit as to how much you need to achieve a proper pregnancy. You don't know about the blastocylation rate, ploidy status, all of this comes into effect. But some papers say at least collect 15 to 20 eggs. But how many cycles do they need for such advanced age group women to get 15 to 20 eggs? Are they prepared? Are they all of this has to be done with enough counseling. And one more thing, being in Chennai and experienced 2-3 rounds of severe floods, we have done oocyte freezing, zygote freezing, and because we were not able to support power and thing because of water getting inside the generator room and all that. So we have achieved pregnancies with zygote freezing, thaw, taking it to blast, and we have had positive pregnancies during 2015, 21, 24, 3, all the floods we've had, all the stages happen also. So, we should be prepared for anything. You never know what you're going to have on that day. So,
I would like to say, Amita, thanks for using jet freezing to salvage the flood.
No, we all go to exchange.
You can't know. We have walked in hip level, chest level water and gone inside the hospital.
There no power. There no power. Yes.
We all
because you never know when the power will get cut off, because our generator room always gets flooded inside. So the maintenance people will say we're going to shut down the power, come soon, and we have to really rush, take a boat and all and go. We have done all that.
Yes, we have seen a lot of videos in Baby Makers group. Thanks for that.
Yeah.
And I would like to ask Charu sir, because Charu sir is running a very successful tip bank.
Sanket, Charu is not there. He's left. He is somewhere else. So he's gone. I think he's on phone.
Okay, sir.
So there is one question related to this cryopreservation. So suppose there is 20 oocyte retrieved, and how you plan your oocyte freezing? On one time, you expose all the oocyte to equilibrium media, or you will do 3-4 at a time?
Like you're gonna divide and do stepwise, or you're gonna take all together and you're gonna freeze them together.
Exactly.
Any experiences from the panelists?
Yeah. Because
it should be done stepwise.
So how many egg you will
Because
keep?
all the oocytes, if you take in one drop and then you get it will be very difficult to do the loading of the oocytes and the to do the time management. 30 oocytes at a time in one drop, and obviously the media volume of the media drop also matters, right? So it should be done stepwise.
So to cut down the time, sir, what we can do is we can divide it like we don't have to wait for complete load till loading. We can divide it. We can divide it likewise. So we can cut down the time also, and we will not be compromising anything from our end also.
Yeah, that is the thing actually. And what about the if you are having that like 30 eggs, so how you plan? Like you will prepare another patty dish for that vitrification, or you will use same patty dish?
Another one.
Another one.
Another one.
It will be expensive.
So actually, you know why
But
I'm asking this. People are doing like that way. Same patty dish they are using for 30 oocytes. They are using same plate and same media. Same. That's why I ask you people just throw that light on that, because people are listening. There are some juniors are also listening these things. They want to know these basic things also. So Sanket, next. Where is Sanket?
Sanket also left.
Yeah, some of the question. No, I'm very much here. So, some of the question, yes, we use a lot of different osmolarities with respect to vitrification and thawing. Say, if you consider culture medium, there will be like stagnant flow of the like osmolarity, where you can say milliosmoles like 280 outside as well as 280 inside, approximately 280 inside. And with respect to ES, equilibration solution, it is almost like 2700 times, like 2700 times, like more than, like at least 10 times more than what you get to see in the inside of the cell. And with respect to VS, it is like 20 times more what you get to see like from the inside of the cell. And something similar to this warming solution, like at the time of thawing. Of course, now we all very much adapted to the universal freezing and thawing protocols, like where you get to see like everything is standardized, like what is the concentration or milliosmoles concentration, what you get to see in WS, DS, and the washing solutions. Like washing solutions are nothing but your buffer medias, where you get to have the equilibrium like milliosmoles of like approximately 280 outside as well as inside, which enables the cells to expand. And of course, as the panel said, like a lot of prime considerations that will be considered with respect to oocyte freezing, because the oocyte freezing is very critical step, and even the thawing, and lot of this considerations should be taken into consideration in order to get the better outcomes. And yeah, I found this very interesting paper where they showed a lot of effect on different extreme factors of the DNA with respect to the freezing. Like when they go under this physical and chemical changes, like fluctuation of pH, temperature, or generation of ROAs with respect to specific kind of cryoprotectant, like say EG, DMSO, PG, and glycerol, and even the physical aspects like hyperosmotic stress does have an impact on the DNA at various stages with respect to genetics as well as epigenetics. And yeah, the last question, I would think, because already we are overshooted the time, it's already 8:30. So quick comments on the effect of stress on oocyte development. Stress in the sense like, example, the female that undergoes like ovarian stimulation, the stress which is going to impact on the oocyte quality, or the normal stress which is going to impact on the oocyte quality. It could be any stress. It could be handling stress at the time of in vitro manipulations also.
So psychological stresses increases cortisol levels, which in turn affects our hormone levels, and that will affect the oocyte quality and the number, as for some of the studies. Other stresses like culture system itself, pH, temperature, osmolarity, light, everything, age, genetic factors, environment, the dose of gonadotrophins, everything has an effect on the oocyte development. So there's just multifactorial.
There is also like in vitro and in vivo stress. In vitro, which is already there, like Dr. Amita said, that there are so many things which are already involved and which can induce stress, like the ovarian stimulation, how you're doing the pickup, everything, the age of the patient, the BMI. And there is in vivo stress, the iatrogenic things, what we are doing inside the lab, how we are doing it, how we are handling it, the temperature, light, culture system, everything is involved. So these all are the, yeah, in vitro and in vivo sort of stress what we have.
I think stress-free is the ultimate goal, holy grail for all our culture systems and what we want to achieve as such. I think we are yet not there, quite far from it, but it should be stress-free as far as possible for the optimal development. And it should be something like the line from, I think, Munna Bhai, say, Bhai tu stress mat le, padhai pe dhyan de. So something similar, we should not give stress to the oocytes or maybe the female, including the psychological stress, which definitely has a negative effect as such. So.
Yeah. Anybody else would add anything for this? The lifestyle factors like alcohol consumption, smoking, any lifestyle habits.
So I feel they all lead to production of ROS and causing stress to the follicles, ovary, and overall development potential of the oocyte. And as Dr. Amita was saying, the cortisol levels, once the cortisol levels are high, they are going to impact on the granulosa cells, which would lead to reduction of production of estradiol, which eventually will not give you the desired oocyte quality for making the best blastocyst. So in all, stress definitely causes harm to the entire process of IVF.
Well, so it's all take-home message for the patients as such, because no matter what, if you undergo stress, we can't do magic at the lab. So it's always about you also, which is going to be equally contributing to the overall gamete quality. So having said that, so these are the studies like different animals and different studies which have been contributing to stress, and of course we can skip this question because already we are overshooting the time. And to sum up. Yes, as we discussed, a lot of points, and take-home message would be like, yes, oocytes are delicate in nature, and the delicate nature of oocytes is highlighting their vulnerability to various environmental and procedural factors during handling and manipulation, especially micro manipulation. And precision as well as expertise also is a take-home message where the stress is most important factor with respect to precision and expertise, where it comes into play, where the oocyte handling techniques has to be minimized to damage the oocyte and maximize the success rates in ART. And also the quality control, which advocates very like rigorous quality control measures throughout the oocyte handling process, including proper monitoring of environmental conditions and equipment calibration and adherence to the strict established protocols. Finally, ethical and legal considerations, we have the advent of law, that is very much important, that we should address all these considerations surrounding oocyte handling, including informed consent, respect for the donor, like autonomy, and responsible use of oocytes in clinical practice and research. Thank you. Thank you, one and all.
All of you, all the panelists, and thanks, Sanket, for making this slides. I know I have nothing. From my side, I'm just sitting. I was not contributed anything in these slides. All slides he prepared himself. So thank you all of you, and thank you for joining. And it was wonderful, excellent panel. And I hope everybody has learned something, and we each other learn newer things. And definitely we will continue with that second part also from Oocyte. And there is one announcement again. I know we already announced these things, but just want to share that this thing. This is the micro embark we are going to do in 8th and 9th June on embryo ecosystem, decoding the science of human embryo culture. Basically, we are going to micro dissect the part of embryo culture system, each and every aspect, and there will be international faculties are also coming. 4 international faculties are also coming. This is our scientific committee. I think 3-4 are here already, and these are also, and we are working hard. Daily we are doing meeting to prepare very good program, and I think we all should attend this program to learn many more things. And this the faculty Nakati Findikali is from Belgium, and there are one more faculty. His name is Alejandro Bartiloki. He is coming self-sponsor. We are not sponsoring anything to him. He want to come just to give a lecture in India. So people are like that because we are doing this organization. IHERA is totally non-profit organization. We are not earning anything. Just we are spreading the knowledge. And I am thankful to my team. They are working very hard day and night. And of course, you will love this program. These are the confirmed national faculty. All are confirmed, already confirmed. And maybe in next month, till 15th, we will be able to uh, uh, prepare the program and share with you all people. So of course, because we are doing that international program, so it will be maybe will take some fees also. So this is very minimum fees, 3,500 plus GST, and PG student it is 2,950 only plus GST. So you all are welcome and to join this meeting and learn from each other. Thank you so much. Thank you. Thank you, everyone.
Also, sir, I would like to add something for here. And
Yeah.
yes, sir, as you said, we're going to come up with the second series of this webinar where we're going to deal about the bioreserve. What does the oocyte contains? Like, what is the reserve with respect to the protein, transcripts, mRNAs, non-coding mRNAs, everything. What is it is equipped with before fertilization? So that would be our next topic of discussion in the next webinar. So thank you so much.
Thank you. Thank you. Thank you, Intas, for helping us. Thank you, everyone.
Thank you.
Thank you, everyone.
Thank you very much.
Thank you,
Much
sir.
all the
Thank you.
participants for joining today and sharing.
Thank you, Ved sir and Sanket for a wonderful for the wonderful discussion.
Thank you so much. Pleasure.
And congratulations, Sanket, you have made a wonderful PPT.
Thank you so much, ma'am. Yeah. Dorai sir, you want to say anything?
Yeah, thank you. Thank you. It's nice presentation. It's actually, it's needed because now the new population is entering to the field. We are the old population. We have to show the right path to them to preserve the legacy of embryology. It's our duty. I personally felt, I think Ved Prakash is taking forward that, taking ahead. It's a wonderful journey. Being a part of the journey, I'm very much happy. So small things, but it will make a large difference in the upcoming population. Actually, as we all seen how the population is coming, the last Parag written on article, no, there is 21 colleges are there. Each colleges around 30 students. Imagine the output of the per year.
Yes.
So it's around 500, 600, minimum 1,000 people are entering to the field every year. So they are the future embryologists. So we have to guide them in the right way. I think Ved Prakash is the right job. Thank you, sir, to take some initiative to guide them in better way.
You know, Dr. Durai, actually, I always say to all my juniors or even to my seniors also, whoever is coming to you, like junior is coming to you, just train them. Don't hide things from them because they are the future. Because they are coming. You will go without saying anything, without teaching them how they will prepare, how they will do the culture in the IVF lab. You don't know. Whatever they learn from our side, they will do the best thing. So it is our duty and it is our responsibility also to teach them in a right manner so they can do better. Our aim should be not to get the pregnancy. It should be get the life healthy baby.
Sure, sir, sure.
So basically, Ved sir, you want to say something like pass on good genes.
Yeah, pass on good genes. You are right.
Good.
Thank you so much, all. Thank you so much, Dr. Akash, for joining at the last moment. And,
Thank you, everyone. I always like to be it gives immense happiness whenever you do a talk or thing. The main thing what I feel is like Duresa said, we might be teaching them. We are probably the link. I can't say we are the seniors over here. So we are probably the link between the older generation and the next generation. We are the bridge over there. But I always feel that when we are preparing for this, we also learn something, and we definitely take home some points whenever we attend these things. So definitely something to learn from everyone, not someone, but everyone.
Everyone.
Thank you.
So much, Amita. Thank you so much, Nancy.
Thank you so much.
Thank you so much for joining us.
Everyone.
Thank you so much.
Thank you.
Thank you so much.
Are we offline?
No, sir. We're still online.
Please take us offline.
Akash sir, your presentation was so good.
Seriously, I thought